If you're about to teach Required Practical 1, which is all to do with enzymes for A-Level Biology, then I'm going to go through it now in this video, talking about the equipment, the method, the results you might collect, and the key things, common exam questions linked to it, mistakes students might make so that you can support them as best as possible. If you are new to my channel, then hello, I'm Miss Estruch and I've been teaching since 2009. Make sure you hit subscribe so you don't miss out on any of these Required Practical videos. And if you want the guides for free, then click the link in the description and you can download these for every single Required Practical. And a big shout out to Woodford County High School. Thank you so much for letting me film these videos here today. Now, this Required Practical is the most vague of all of them because it just says investigate the effect of a named variable on an enzyme required practical. So there isn't an exact method that you need to know for this because it could be any of the variables that affect enzymes and any enzyme controlled reaction. And for that reason, you don't usually see in the exam a question that is noticeably visible as Required Practical 1. And instead, they're more interested in the apparatus and techniques that come up in this practical. So I'm really going to be emphasizing that as I go through the transferable skills that could come up linked to this practical in the exam, whilst also applying it to this particular method. But the chances of in an exam it being literally this method are very unlikely, and students don't have to memorize the equipment and the method. It's all about understanding it and linking it to apparatus and technique. So let's have a look at our equipment to start. So the equipment that you're going to need for this experiment is, I'm actually going to do pH as the variable that I'm changing, and we are using trypsin, and we are going to be looking at how long it takes it to digest the gelatin in our photographic film. So that is our substrate. The enzyme is trypsin, and we are changing the variable of pH. I've got 3 pHs that I'm going to be testing today, but if you've got access to more, then you can have a wider range. And in particular, if you've got smaller intervals, that's going to give you more accurate results as well. I don't actually have the boiled enzyme, which you can use as a control. Instead, as a control experiment, we're going to be using distilled water. I need to measure out 4 centimeters cubed of my buffer and my enzyme. So I've got 5 ml syringes so that students can practice accurately measuring with a syringe. I'm going to be controlling the temperature. So I've got a beaker and a thermometer. So I can use this to create a water bath. And I've got my thermometer so I can continually check the temperature hasn't changed throughout. And if it does, add more water. I've also got my test tubes and a test tube rack so that I can keep them in this securely and safely when I'm not using them. I've got a stop clock to record the time taken. And then lastly, I have got my photographic film, and these are attached to straws so that I can dip them in and out of the enzyme solution to check how long it takes for the film to go completely colorless. So the risk assessment for this Required Practical. First of all, we are using different pH buffers and we're using our trypsin, both of which could be an irritant. So if you do get it on your skin, wash your hands straight away. And we're going to be using goggles as well to protect our eyes. To create the water bath, we are going to be using a kettle of hot water. So students need to make sure they're handling that with care to prevent scalding themselves. And then the most common thing that I see happen in this experiment is breaking glassware, particularly when it comes to using test tubes, because students might just leave them on the table and then they roll off and smash. So really emphasize to the students that when they're not using their test tubes and they're not inside of the beaker, they have to be in the test tube rack to reduce the likelihood of them rolling, smashing, and them cutting themselves. So one really common exam question that comes up with this Required Practical, or at least linked to enzyme practicals, is the idea of a control experiment. Students need to know that a control experiment is when you do the exact same experiment again, using the same equipment, the same method, but you remove whatever you think is deliberately causing the change. Say for this experiment, we think the enzyme is what is causing the reaction to occur, or at least it's catalyzing the reaction. So that means we would need to do as a control experiment the exact same method, exact same equipment, but without the enzyme. And the best way to do that is actually use the boiled version of that enzyme. So you've denatured it. And that's actually better than using the same volume of distilled water because it proves to you that it's nothing else about the enzyme, potentially the fact that it's a protein, that is causing the change. And it is the enzyme and the active site in particular that is the cause of the changes. So that's one key thing students need to know for the exam. The control experiment, keep everything the same method, but use a boiled enzyme. They might be asked why they have to use a control experiment, and that is so that they can compare the control experiment results to the test results results. And that helps to prove that any changes that we see happen are due to the enzyme and not anything else in the investigation. So I'm now going to create my water bath, and ideally you want your water bath to be at around body temperature, so 37 degrees C, or you might say 40 degrees C, because this enzyme trypsin, that is naturally occurring within the body, so you would think that the optimum would be around body temperature. Now, it doesn't matter what volume of water that they're using in the water bath, as long as you have a large enough volume to cover the test tubes, but make sure the students don't fill it all the way to the top, because then when you put the test tubes in, that is going to cause it to overflow. So I tend to tell them to just half fill it at this stage. Now, if we want it to be about 37, I actually boiled that kettle a while ago. The temperature of the water from the kettle does drop quite quickly from 100 to 90, but still, we want this at around 37 or 40. You decide. I'm going to go for 40 degrees C. So I've deliberately not half filled it because I know that I'm going to have to add some cold water to bring the temperature down a bit. So let's have a look at the temperature of this water at the moment. So actually it's not far off because we boiled that quite a long time ago. This is at 50 degrees. So I'm going to add a tiny bit of cold water. Okay, that's gone down to 47. 45. Okay, so that is at about 42, but I'm going to leave it at 42 because I'm not quite ready. One thing to point out to your students is we've made the water bath, but that temperature is going to continue to drop. So they need to continually check the temperature throughout the experiment. And if the temperature has decreased, they'll need to add some more hot water to bring the temperature back up again. So that's my water bath, and that is to control the temperature to make sure it's only the different pHs. That is the variable we are deliberately changing. That is our independent variable. This is our control variable. Now I'm actually going to label my test tubes before I go any further to make sure I do put the correct thing in each. So for my distilled water, that's what I'm using as my control experiment. So I'm going to label that as C for control. I've now got pH 7, 4, and 9.2. So I'll label as 4, 7, and 9.2. And these over here are just spares. So I'm now going to label my test tubes, and I've actually labeled 3 of them already, pH 4, 7, and 9, and those are going to be my test ones. But I also need my control experiment where I don't add the enzyme and I add the distilled water. So for this one, I'm going to call it C for control and 4, C for control and 7, and then C for control and 9.2. I've also got a separate syringe for each of those buffers and a separate one for the trypsin, because one common mistake that students will make is cross-contaminating, and they might accidentally swap the syringes they use and they don't necessarily have the exact pH that they think, and we don't want to be mixing the enzyme into it either. Okay, so that's the next thing I'm going to do then is add 4 centimeters cubed of each of these to those. And when they're using the syringe, make sure the bottom of the plunger is at the black line for 4. So 4 centimeters cubed. Okay, so we now have the buffers in. And I'm actually going to put those in my water bath so that those are going to reach the temperature of this. And at the same time, I'm going to keep my enzyme in a separate water bath as well, so that that's going to be at 40 degrees C. Take those out. Okay, so now I'm going to add 4 centimeters cubed of the enzyme only to the front 3, which were our test ones. The back 3, I'll be adding 4 centimeters cubed of water, not the enzyme. Okay, and the last one is, this would either be your boiled enzyme that you'd be adding, or I'm adding distilled water. Same volume, to make sure it's nothing about the volume causing the changes. So now all of our test tubes are ready. We're going to put it into the water bath. And as we can see, the volume of the water in the water bath has gone up a lot because we've added 6 test tubes to it. We now need to leave these so that the contents of the test tube reaches 40 degrees C, which would probably take about 10 minutes, but students would need to put a thermometer inside of the test tube to confirm that. But each time they then need to clean the thermometer before they put it into another one so they're not mixing the contents. So this experiment is the enzyme is breaking down the gelatin in the photo film, which is what makes it black. So when it goes completely clear, which it nearly has now, that means it's all broken down, but we're using different pHs to show that pH effects. Now, while that is acclimatizing, one other key thing just to be aware of in general, in terms of techniques and anything linked to experiments, that color change is the endpoint, which it is in this one. The endpoint of this experiment is when this piece of film goes colorless, and you might not even be able to see it. I'm not sure how well you can actually see it, but that is just a clear and colorless piece of photographic film. So what we are investigating is the time it takes for this photographic film to lose its color and go completely clear. And that's because it contains gelatin. That is the substrate. Trypsin is the enzyme that's going to catalyze the breakdown of that. And we are investigating it with 3 different pHs to show that pH does have an effect on enzyme-controlled reactions. But we are measuring the time taken for that film to go completely colorless. And because that is subjective, when I think it's completely colorless and clear, it might be different to someone else, what you should do is have a piece of film that is already completely colorless and clear to compare it to as your standard. And that helps to standardize the method. So that's what we are doing here. That's why I've got these 2 bits at the ready. We will also need a piece of film to go in all of these test tubes. And when it has reached temperature, I'm then going to put them in, start the stop clock immediately, and I'm going to time how long it takes them to go colorless. The controls we are not expecting to change. And we've got pH 4, 7, and 9.2. So we might expect 7 should go the fastest because that is neutral. Now this one in particular, they could also comment when it comes to their write-up. We can see that these pieces of film are not exactly the same size. So that means our substrate volume and the concentration should be the same, but the volume of the substrate is different. The larger pieces of film will take longer to turn colorless than a smaller piece. And actually this, I might find that some of them fall off. So we'll see how it goes. Let's just check first of all that it is at 40 degrees. So my water bath has actually dropped in temperature down to 28. So let's add some hot. Another reason why I say only half fill it, because you're going to need to top up with hot water throughout. Okay, now this bit is easiest if you have 2 people so that you can do this all at the same time. Otherwise it's going to be inaccurate because I now need to get all of these in and start the stop clock. So this is where I do think working in pairs is helpful because I'm just going to have to quickly get those in and start my stop clock. And this one is going to have to be on a separate stop clock because it's going in slightly after. So this one's going in 10 seconds after. And now they need to consistently, to standardize the method again, move the sticks so they all get the same level of mixing and checking. So I'd recommend that they just go round in a circle like this, and then that's approximately 5 to 10 seconds. Okay, and we do need to be quite quick on it because we can see this one. Let's compare it. Okay, so that one's already gone. So 1:46, 9.2. So that's our first one. And then the students will need to record down that time. So this one looks like it's nearly done. So this is pH 7. So I wouldn't say that is completely clear. So put it back in. Okay. So I'd say that one is done. So that's our pH 7, 327. Which means we now only have one left that we think is going to change. Now you can see at this point, if students are writing down rough notes like this, that's absolutely fine. But I'm going to project up here an example of a table that you could do. So you always have to have your independent variable on the left-hand side, dependent variable on the right-hand side. And I would go for our units in seconds. So 1 minute, 46 seconds. I would need to change that to 106 seconds. And for 3 minutes, 26, that would be 407 seconds. So you'd need to convert it so you've got the same units throughout. If you do have time as well, they could do 2 extra repeats. You've got 3 trials because then they can identify anomalies to exclude from the mean. And again, just check your temperature throughout. Okay, and then last one, 8.28, and again, we'd then need to convert that. So that would be how you would do this particular example of the enzyme required practical. Then you can get the students to pull their class data, calculate a mean, identify any anomalies, and then they could plot a graph of the mean, time taken, or you can convert it into rate of reaction with those different pHs, so they should get that typical curve that you'd see with pH. And as a reminder, if you want to download for free an example of an experiment you could do for this practical, then I'll link it below. If you found this helpful, don't forget to subscribe, so I'm doing all of the required practicals, and for the document that has the method, the plan, risk assessment, common tips and tricks, and the results for this one as well, then I'll link that in the description below. In this video, I'm going to be going through the root tip squash required practical for A-Level Biology. We're going to be looking at the equipment, the method, and then tips and tricks to try and help make sure that it works. Because if you've ever done this before, you'll know notoriously it's so difficult for students to be able to find any cells that are currently undergoing mitosis. If you want to download for free this practical guide that has everything that I'm going to go through in this video, then I'll link it in the description below, and make sure you are subscribed because I'm going through every single required practical. Plus a special shout out and thank you to Woodford County High School for having me in to film these practicals. The whole aim of this practical is we want students to be able to identify cells under the microscope that are currently undergoing mitosis, so they can see and hopefully identify the different stages of mitosis, so what they learn in terms of the behavior of chromosomes during the different phases of mitosis, they can actually see on the microscope. Plus it links to that math skill, the mitotic index, where they need to be able to identify how many cells there are in the field of view and how many cells are currently undergoing mitosis, and then they can work that out as a proportion. But before we jump into all of that, let's have a look at our equipment. So you are going to need, I'm using garlic today, garlic that's been left to grow, submerged in water, so you do have roots. We need acid, and the role of this acid, which is actually a really common exam question now, is that is going to stop mitosis happening any further, so we get a snapshot. It also helps to break down the connections and the links between the cell walls so that when we squash it, we do get a single layer of cells, and it can also help with the stain being absorbed into the cell and the chromosomes getting stained. That takes us on to the stain, and I'm actually using toluidine blue for this, and this will stain the chromosomes so that they become visible when we look under the microscope. We have our cover slides and our cover slips. I've got a stop clock here because some of the stages we need to leave to happen for about 10 to 15 minutes, but they don't have to have accurate timings for that. I've got a mounted needle, and I've got some sharp scissors, but they could also use a scalpel cutting onto a white tile. And for the heating stages, I'm actually going to opt to use this heating mantle, this heating plate, because it's far safer in terms of not having hot water that could scald and spill, and not using a naked flame. But you may not have this, so you could opt for using a water bath instead. So the key things that you need to be aware of in terms of the risk assessment are, we are using 1 molar hydrochloric acid, so you have to have eye protection on at all times, and throughout I'm actually going to be using gloves at any point when I'm using the acid as well. The stain can be an irritant, plus it can just stain your skin and clothes, but that's not so much a risk. But we are wearing goggles because it could be an irritant for the eyes. We then also have our slides and our cover slips, which are glass. They could break and cause cuts, in particular the very, very fine cover slips. So do make students aware that they are very, very thin, and if they push down too hard at one point rather than an even pressure across a larger surface area, it could snap and shatter the cover slip. But I'll show that when we get to that point as well. We've got the sharp mounted needle, sharp scissors, and also the scalpel would be sharp, so students need to handle those with care, and any cutting needs to be on a hard surface away from them. And then lastly, depending on what you're using as your heat source to heat up the acid, students need to be careful that they're not going to be burning themselves. If you're using a heating mantle, pointing out that they shouldn't touch the top. If it's hot water, making sure that they are handling the kettle, if that's what you're using, with care as well. So in terms of the variables for this investigation, it's not really one where it's a quantitative experiment that we're getting a quantitative result of an endpoint of reaction. We're more interested in seeing the actual stages of mitosis. However, there are things that we could control if we were going to compare maybe two different plants, for example. So we need to make sure that we are going to be using the same age garlic each time. Ideally, it'd be from the same garlic, same age. We'd need to leave it in the acid for the same amount of time. We'd need to use the same volume and concentration of our indicator as well. So the first step I've actually already done and got set up because I was doing this earlier, but just to go through what you'd need to do. I've got the garlic, and using my sharp scissors, I cut off just the very, very tip of that root. So it was about 3 millimeters from the end, and I took 3 root tips and I placed them onto this watch glass here with a few drops of acid, and it's been heating on this heating mantle at about 50 degrees for over 15 minutes. I also used the mounted needle to help macerate it, and all of this is to help break down the cells that are connected, so when we come to squash it, we do get a single layer of cells so that light can pass through and we'll be able to visualize the results. Acid also there to stop mitosis, as we talked about earlier on in the video. So the next step then is I'm going to add some stain to this while it is heating as well. I'm going to leave that on the heating mantle with the root tips for about 2 minutes. And this is to give a chance for that stain to get into the cells, get to the chromosomes and stain them. So while I'm waiting, I've got my slide and I'll get my cover slip as well. I'm just going to remove some of the excess stain. Let's take these gloves off. Rinse it with some water just so I can see. Right, I'm going to try and deliberately now just suck up my root tips and place them onto my slide. So there's some of it there. Just going to get some of that excess liquid. And there's some more root tip. And then I'm going to move, this is like my main root tip that I'm going to aim to see. So use my mounted needle, just position that there. Place the cover slip on top. And now this is where we get the squash part. So I'm going to put a piece of tissue paper on top, and we need to squash so that we do get that single layer of cells, but we need to make sure we're not just pushing down in one place because that could shatter the glass. So I'm going to place this on top, so it's also going to absorb any liquid either side, and just smooth my hand, not sliding it because we don't want to slide it. I'm keeping it in one position, but just going back and forth. So it's going to gently squash it. And now we can see that we have got our root tip squash slide created. So now we've made our slide, we're going to mount it onto the optical microscope and see, can we actually find any cells undergoing mitosis? I would get the students to clear away everything that could be harmful. So clear away the stain, the acid, and all of that at this stage. So we now just have our slides. They can take off their goggles looking at the microscope. So I'm going to take my microscope slide and get the students to place it on the stage. and it's then clipped into position. Always start with the lowest power magnification first, because it's easiest then to find all of the cells, and then they can work their way up. And depending on what type of microscope you have, it may be the same level of sophistication, this or not. I've actually got a dial here that I can turn to move my specimen so I can see it's directly under the light, so I know it's going to be in the correct position. So I'm moving it so I can just see that is now in the right position. Then have a look through. And I just need to focus it a bit, and then I'll show you what I'm currently looking at. So that's what I can currently see, and I would get students to take video through the lens or photos through the lens so that they can easily describe or point out to you what they can see. But at this stage, the cells, we don't have anywhere near a high enough magnification to actually see the chromosomes within the cells. So now I've actually got it in the correct position, I can move up a magnification. So for context, that was my 4 times magnification lens, and now I'm going up to my 10 times. So I can still see, but I'm just going to slightly change the position so I can see a section where I think hopefully we'll get some good cells to see. So again, I'll just show you what I was looking at. We're gonna move up to a high magnification this time, times 40. Do let the students know that they need to be careful that each of these lenses is longer, so they need to make sure that they don't accidentally move the lens down and crush your slide. So that's why I was actually just checking from the side, making sure there was space. So then I'm gonna have another look, and I was using these dials here, which are like our coarse and fine focus, to get the image into focus. If students are struggling to get it to focus, generally speaking, it pops into focus when the lens is very, very close to the slide. So if there's a big gap between the lens and the slide, then it's probably not going to focus. It needs to be really close to it. But as I said, check that they don't then move it all the way down and it crushes and smashes the slide. So this is now in focus, and I can see cells. I'm just going to move it around. Okay, I think I can actually see some already. It might be in stages of mitosis. I'm just going to adjust the focus and then take a photo so you can see what I'm seeing. Now at this point, it's worth noting, and this is what's happening to me, and just to tell the students, because you're now at such a high magnification, even the slightest change to your focus dials can very quickly make it go out of focus. So you want to be using the fine focus, not the coarse one at this stage. I've actually got one here that goes up to 100 times. So let's just move that in place. I've actually lost the position of it now. Let's get you back. Oh, it's over there. Okay, so go this way. And then if I go backwards, it should be bang under now. Yeah, there we go. I suppose I should say that's a tip rather than just saying it out loud. So what I was just doing then was, because I took the slide out to put the oil on, when I put it back, it must have been at a slightly different position. And when you're at that high magnification, it can make it really difficult to find the cells that you were looking at. So what I then did was just moved it all the way out to make sure I definitely had it aligned in the correct position in this way. So let's call that vertically. So when I moved it backwards, I could see when it went exactly underneath the lens and it would be in the correct position. And now I have managed to find it without having to go all the way back up to the lower magnifications. So now it's just focusing. So the hardest bit now is, so I've got it in focus and I can see all the cells, but this is when students now need to have quite a bit of patience and just move along their slide and try and find a field of view that has any cells undergoing mitosis. So that's what I'm currently doing. There's some that look like they might be in telophase, which I'll show you, but I'm just going to keep looking to see if I can find, because if there's a metaphase or an anaphase, that's a lot more obvious visually. You can see one section here where I've not squashed it enough because I've got several layers of cells, so I needed to squash that a bit more. Now, one of my top tips is always have a pre-made slide or image that you can show the students so they know what they should be looking for, and hopefully they've got enough time to be able to look and find something. So I'm actually going to show you here one of the photos that's actually come from one of my colleagues who got this image when she was doing this practical. So here you can see clearly the cells in the different stages of mitosis, and this is the sort of thing that hopefully the students are able to find. Now, it's always good to have a backup in case yours doesn't work. So I'm actually using here a slide that my teacher friend, Mrs. Egan, managed to get really good results for when she taught this recently, to demonstrate how your students could then collect their results. So either they'll get an image like this themselves, or if not, I'd recommend you provide them with an image that they can then calculate the mitotic index on. And the mitotic index is when you're doing the number of stages visible that are in mitosis divided by the total number of cells visible. And I'd recommend to the students that they have a look, take a photo, print it off, and then work it out. Because if we go through this one, it's going to be really hard to count all the cells without losing track unless you can cross them off. So the first thing that I would do then is work out which cells are in mitosis. And what you need to tell your students for this is, any cell that there are visible chromosomes in is in mitosis. So all of these where you can't see the individual chromosomes, it looks a bit like a blurry, shady part, that would be interphase. Whereas this here is a cell in mitosis in anaphase, because we can see the chromatids have been pulled to opposite poles. This one here is in metaphase. We can see those chromosomes lining up at the equator. And we've got another one up here in anaphase. As for all of the rest, I can't see any others where there are visible chromosomes. There is one here, but because the cell is half included, half not, you wouldn't include it in your count. And I would give that as a general rule to the students. Unless it is an entire cell in view, do not include it. That is where I've gone through and counted all of the complete cells. So these I didn't include as completed cells because you can't see the whole thing. And I had 47. And then I've got 1, 2, 3 cells that are in mitosis. We've got anaphase, metaphase, anaphase. So to calculate the mitotic index then, we would need to do 3 divided by 47. 3 because we've got 3 cells in mitosis, 47 because that's how many cells I could see. And that comes to 0.06. That would be the mitotic index. And then you could encourage students to pull their class data if they have done this using the same garlic or the same species of garlic and work out an average. Or even better, if there's time and they've got good results, they can then look at another field of view from their own microscope slide and calculate an average. So that is it for the mitosis, a root tip squash required practical. Remember, if you do want to have this entire method, practical top tips and the guide for free, it's linked in the description, and make sure you're subscribed so you don't miss out on the next practical video. In this video, I'm going to be going through the required practical 3 for AQA A-Level Biology, which is the osmosis practical, and I'm going to be using potato as my plant tissue. We're going to be going through the equipment, the method, common mistakes students make, common exam questions, the actual experiment itself, and how to interpret the results. And if you want all of that bundled together in a free guide, I've got it linked in the description so you can click just there to get your hands on it. I've got a video like this for all of the required practicals, so click subscribe and you'll be notified as soon as each one comes out. And a massive thank you to Woodford County High School for having me in today to film all of these practicals. So the aim of this required practical for students is they need to work out what is the water potential inside of a piece of plant tissue, and also learn how to do a dilution series. And they're going to be working out the water potential inside of the plant tissue, or in this case, the potato, by putting the potato in different concentrations of sucrose. So we can then work out at which concentration of sucrose do we get 0 change in mass. That indicates the concentration of sucrose in the potato chip. And then we can look up what water potential that would be. So the equipment that I've got then, I have got pre-cut for me 6 potato cylinders. I've got 6 boiling tubes. I've got a range of smaller beakers and also my distilled water and 1 mole of sucrose solution to start with. Those I'm going to be using to create the dilution series, as well as these different size syringes. I've got a 10 milliliter and a 5 milliliter syringe. I'll be using those in my serial dilution. I've got a large 500 milliliter beaker here and a thermometer, which will act as my water bath. I've got my boiling tube rack for my boiling tubes, stop clock, tissue for blotting, kettle with the hot water for the water bath. I've got the bung to put on for the mixing in between the serial dilutions, forceps to get the potato in and out of the boiling tubes, white tile and scalpel for the cutting. And then I've also got a ruler so that I can measure the initial length and final length, as well as I've got my balance here to measure the initial and final mass mass of each potato as well. And lastly, I've got my permanent marker to label all of the boiling tubes so that I don't confuse which potato has gone into which boiling tube. So the different variables in this experiment, they're independent variables, so we are deliberately going to be changing the sucrose concentration. And we'll need to do that first. We'll need to create our dilution series to get those different concentrations. The dependent variable is going to be our percentage change in mass. So we will need to measure the initial mass, the final mass, and then work that out as a percentage change in mass. And then our control variables, we will need to make sure that all of the potato chips are placed in a water bath at the same temperature because temperature affects the rate of osmosis. We also need to have the same size and shape potato because the surface area to volume ratio will also affect the rate of osmosis. And then also things like the length of time that we leave each potato chip. Also the species and the age can have an impact as well. So let's think about the risk then. So we can come up with a risk assessment to make the students aware of if they're not having to research it themselves. So first of all, we can see over here that for those potato chips, these ones have actually been prepared already, but you could use a cork borer, which is quite sharp, and you need to make sure you don't have any skin on either side. And for that, we could use a scalpel on our white tile to cut it. So the risk here would be the blade, the scalpel could cut us. So students need to have a sharp blade, cut onto a hard surface and cut away from themselves to minimize the likelihood of cutting themselves. Next, we have a lot of glassware in this experiment, particularly the boiling tubes, because they could roll off and smash. We need to make sure that students are handling them with care, and when they're not using them, they're going into a boiling tube holder, so they're less likely to roll and smash. I'm also going to be using some hot water from the kettle to create my water bath, so students should be handling the hot water with care to reduce the likelihood of scalding themselves. So my first step then is to create my dilution series. So we can see here on the table, these are the different concentrations of sucrose solution that I'm going to be making. 0, 0.2, 0.4, 0.6, 0.8, and 1 moles per decimeter cubed. And we've got the different volumes there in the table that I'll need to add of my distilled water and of my sucrose solution. So I'm actually going to label my boiling tubes now. So this will be my 0, so that'll be just distilled water. 0.2, 0.2, 0.4, 0.6, 0.8, and 1. So the 1 mole, that's exactly what I've got here. The 0 is the distilled water. So I'm just going to put those in straight away, making sure that the bottom of the plunger is exactly on the line of the 10. I want 20 milliliters or centimeters cubed, so I'm going to do that twice. And then this one is just distilled water. Okay, so that's those two made. The rest now, we are going to need to create these different concentrations. Now, all of these need to have the same volume in, and we're having 20 centimeters cubed. So the students could either do a dilution series, or like the table that I showed, they could work out using the serial dilution calculation to work out what volume of the 1 molar sucrose solution do they need to add and what volume of the distilled water do they need to add. Now, the next one that I've got is 0.8. So I need to have 16 of sucrose and 4 of water. So 16. 10, 16, and then 4 of water to add up to 20, so the same total volume. Then we have got 0.6, so for this one I need 12 of sucrose and 8 of water. Now we have our 0.4, which is the reverse of this one. So we now need 12 of water and 8 of sucrose. And then our final one, 0.2, we're going to need 16 of water and 4 of the sucrose. For the 0.2, we need 4 of the sucrose solution and 16 of the distilled water. 4. Or this one. So I've got all of those, but for each one, I'm just going to place the bung in and just invert to make sure we do have that thoroughly mixed. So we have the concentration we're expecting. I'm just dabbing it each time to reduce transferring over any. So So we've now created our different sucrose concentration solutions. So we said we're going to be controlling the temperature, and I'm going to put them all at about 30 degrees C because that's going to be warm enough that we should get a set of results within the space of an hour's lesson. So I am going to pour it into here. This is boiling water from the kettle. This is probably already cooled down to about 60 or 70 degrees C. Let's see what it's at so we know how much cold water to add. Make students aware though, don't fill it to the top because by the time you've added all of these boiling tubes, the volume of the water is going to rise and it could overflow. Now that is 70. So I then add some cold water from the tap to bring that down to 30. Now, one of the key things to make students aware here is they'll set up the water bath now at this point, and often students then forget about it and they don't consider the fact that the temperature is going to continually drop throughout the experiment. So they should be regularly checking the temperature on their thermometer, and if it's gone lower than 30, they add more hot water until it's up to 30. And that's quite a common exam question now, how you can check that your temperature of the water bath is remaining constant. Right, that's still 60, so I'm going to add some more. Now at this stage, mine's getting a bit too full, so I'm going to pour some out. One beaker seems to drop it by about 10 degrees, and I'm still at 50. Now, depending on what temperature the hot water comes out of your taps, you probably wouldn't actually need a kettle for this because that was obviously a lot hotter than 30 degrees C. The water coming out of the taps may well be about 40 to 45 degrees C, so you might not need the kettle. So that's something that you could test in advance. So I've now got mine down to temperature. And what I'm going to do next then is place my boiling tubes into the water bath. And yeah, even mine there is too full. So I'm going to leave those to all come to the same temperature, which will probably take about 5 minutes, and they need to check that with their thermometer. Now, while that is reaching temperature, this is now when we can get our potato chips ready. So I would suggest they set that up first, and then at the same time, we can be doing this. So I've got my balance, a little tray that I'm going to be placing the potatoes in each time. Mine are actually prepared already. So as I was saying, there was a cork borer that was used to take these slices out of the same potato. So we've got that as a control variable. And then they were cut to the same length. But I'm going to double check that. So yeah, we can see a pretty good job there. They have been cut to basically the same length. But what we can then do is, on a piece of tissue paper, or it could just be a piece of paper, to make sure we don't mix up which is going into which, I would now actually write on my tissue paper that this is the potato I'm going to put in at 0, 0.2, 0.4 concentration, 0.6, 0.8, and 1. I've also got some tissue paper here ready to blot them dry. And again, not squeeze, because if you squeeze, it's going to draw water out from within the cells. We just want to remove any of the water on the outside because that's not actually the mass of the potato. That's just water on the outside. So now I'm just going to press the 0 button so it knows that that is 0 mass with that tray on. Place my potato chip that I've blotted in, and I've got 6.02. You could, if you wanted to as well, get students to measure the initial length and final lengths. We could do a percentage change in length and a percentage change in mass. I'm just going to do percentage change in mass for now. And that is the one that I'm going to place in the 0 concentration. So this is how I'm going to be keeping track of it. And then you need to do that for all of your potato chips. And if you do ever get where it's deviating from 0 when it's empty, just press the 0 button again. In fact, I'm not going to use this one because I can see that there's a bit of a hollow part there. So that means we don't actually have the same surface area as the others. So I'm going to go for this one instead. That's 611. So we've measured all of those start masses. Once it's then gone into the water, we'll leave it for, it depends how long your lesson is. Ideally, you want at least 30 minutes. But if you only have 20, you could always increase the temperature slightly because then you should have osmosis happening at a faster rate. But I'm going to leave it for at least 30 minutes and then we'll come back and see the change in mass. And we're going to do it as a percentage change in mass because we can see here, these are our starting masses. And even though we've used the same potato, same cork borer, cut them to the same lengths, none of them are the same starting mass. So that's why we'll do it as a percentage change in mass to take into account none of them started off the same. So now I have to be really careful. I definitely put them into the correct tubes. So this one is 0, 0.2, 0.4, 0.6, 0.8, and 1. And now I'm going to leave them for about half an hour. Right, so for us it's been, I think, about 20-25 minutes. So we're now going to take those out and measure the final mass. Now I did have forceps, so as you can see, didn't actually fit in. So sometimes it is easy just to have a sieve, pour it into the sink and do it that way. Now we've got our final results as well on a tissue, but that would then need to be recorded into a proper table and plot your percentage change in mass against the sucrose concentration. So while we're waiting for osmosis to happen to be able to actually see any impact on the masses, let's think about what the results would be. So students are going to measure the initial mass and the final mass, and then they work out the percentage change. And this is how students would calculate the percentage change. The equation, percentage change in mass, brackets, final mass minus initial mass, brackets, divided by initial mass. Then we're going to plot that onto a graph to make a calibration curve. So on the x-axis, students will need to plot their sucrose concentration, and on their y-axis, they would have their percentage change in mass. And if they have multiple repeats, or you're pooling the class data and calculate a mean, then that would be the mean percentage change in mass. Students can then plot their curved line of best fit to create that calibration curve. And it's at the point that the line crosses the 0% change in mass, that indicates the concentration inside of our potato chips. And then we could look up for that sucrose concentration, what is the water potential? And in that way, we have used a dilution series of sucrose concentrations, doing the osmosis required practical to identify the water potential of plant tissue, in this case, our potato plant. So in summary, some of the common mistakes to look out for are the labeling of the potatoes to make sure they're going into the correct boiling tube. So doing something like this, where you are literally labeling on a piece of paper which piece of potato is going into which boiling tube and what was their start mass, and make sure you're taking it out and placing it in the same place, and all of these are labeled. That should really help. Making sure students are definitely blotting the potato before it is always going to be weighed, but blotting in a gentle, consistent way, not squeezing it, because that draws water out from the cells themselves and therefore affecting the mass. And also when it comes to creating your different concentrations of sucrose solutions, really check that students are using their syringe accurately, so they're definitely creating the concentration that they think. So that is it for the osmosis required practical. Don't forget you can click the link in the description to download this entire practical guide for free, and make sure you hit the subscribe button so you don't miss out on any future practicals. In this video, I'm going to be going through with you required practical 4 for A-Level Biology AQA, and that is the beetroot practical. I'm going to be looking at different concentrations of ethanol on the permeability of the beetroot membrane. Now I'm going to be talking you through the equipment, the method, how you could then analyze the results, and also some top little tips for success. And if you want that guide completely for free with that information, it's linked in the description below. I'm doing this for all of the required practicals, so make sure you're subscribed so you don't miss out. And a big thank you to Woodford County High School for having me here today. So the aim of this experiment is looking at the cell membrane, that phospholipid bilayer. And you could look at the effects of different temperatures, different solvents. I'm actually just using one solvent, ethanol as a type of alcohol, and varying the concentration. And we're going to be investigating how the different concentrations of ethanol affect the permeability of the membrane, that cell surface membrane of the beetroot. And the way that we can measure this is by looking at how much of that red, that dark red-purple pigment called betulin, how much of that comes out of the beetroot cells into the solution. And the more pigment that comes out, out, that must mean that the membrane, that cell surface membrane, has become more permeable. So we're going to be doing this based on how intense that red colour is. You could use a colour standard where you prepare known concentrations so you can see how intense that colour looks and compare the colour of the two test tubes. That is still qualitative results though, and it's still subjective, even though you have got the comparison to help standardise. So even better would be to use a colorimeter, because that will turn our qualitative results, the description of the intensity of the colour, into a numerical value, which is our absorbance of light through the sample. So that's the version we're going to be doing. So the theory behind this one is ethanol is an alcohol, and alcohols are going to dissolve the lipids, and our phospholipid bilayer, because it's phospholipid, it will be dissolved by the ethanol. So I'd be expecting that the higher the concentration of ethanol, the more it dissolves that phospholipid bilayer, and the more we get of that betulin pigment moving out. If you were doing this with temperature, then the higher the temperature, that would break the bonds in the tertiary structure of the proteins embedded within the membrane. So your channel proteins, for example, and that would then mean the channel proteins might be more open or permanently open, so more pigment can diffuse out. So here is the equipment that I'm going to be using. So we've got the beetroot, which is already cut up into discs. Now, in terms of control variables students need to be aware of, they should be using the same beetroots, and therefore we know it's the same species, the same age, should be the same size and shape discs, but you might want them to measure the mass of each disc before we put it in as well, just to help to have that there as a consideration. I've also then got my test tubes here where I'm going to place the beetroot discs with the different concentrations in each test tube of ethanol, and a larger beaker with a thermometer, because that's going to be my water bath, because I'm going to be controlling the temperature as well, because I only want to know the impact of the different concentrations of ethanol. That takes us on to the different concentrations of ethanol. I've got this as a percentage of ethanol. You could get the students to create these concentrations themselves, but because serial dilutions isn't a focus of this required practical, I'm not going to be. But I do have my 100% ethanol. I then have 80%. I have my 60%, 40, 20, and 0% ethanol. That is my distilled water. And then I've got a different syringe for each of those so we don't cross-contaminate. I have permanent marker so I can label all of the test tubes so I don't get any of those confused. And then I've got my test tube rack to hold the test tubes as well when I'm not using them. Forceps so you can transfer your beetroot discs into your test tubes. And then one of the key things, we have a bung that's going to go on top of all of those. I've got some cling film on as well. I'll come back to that in a bit more detail, why we do that later, but it's to do with preventing evaporation. That's a really common exam question, but I'll come to that when I set it up. Lastly, we can see over here, I've got my colorimeter and my cuvette, which I'm going to be using to turn my qualitative results into quantitative results. And on top of that, I do have an organic waste bottle to pour all the ethanol in at the end, because ethanol is harmful to the environment, so it cannot be poured down the sink. So in terms of the variables for this investigation, my independent variable is going to be the concentration of ethanol. That is what I'm deliberately changing. I'm going to be measuring the absorbance of light through the different samples using my colorimeter. So the absorbance of light, that is going to be my dependent variable. And then control variables. Some of them I've mentioned as I was going through the equipment, but let's just have a solid list so you know what to make the students aware of. Temperature of the water bath, because temperature is going to affect diffusion and also it can affect the permeability of the membrane. And I'm going to be controlling that by using a water bath. Make sure the students are keeping an eye on the temperature throughout, because the temperature will decrease. They might need to add more hot water to make sure it stays at whatever temperature you go for. I'm going to go for 37 degrees C. The volume of alcohol that you actually use. So I'm going to be adding 5 milliliters to every single test tube. The time that the beetroot is left for, so students will actually also need a stop clock in their equipment as well. The size and the surface area of those beetroot discs, because if you've got a larger surface area, then you might have more diffusion of the pigment coming out. And the same species and the same actual beetroot as well, because that could have an impact. So next then, thinking about our risk assessment. We are using ethanol, which is highly flammable, so make sure there are no naked flames. It is also very dangerous if it gets ingested, so students shouldn't be drinking or taking any chemical in the lab anyway, but it's worth pointing out that it is incredibly harmful. And it is also harmful to the environment, so we do have to put all of the ethanol samples into the organic waste bottle at the end. Nothing should go down the sink. Now, if your students are going to be preparing the beetroot discs themselves, then they will need a cork borer and a scalpel, both of which are sharp and could cause cuts. Now, to reduce the risk of that, I've actually got this pre-prepared, but it depends how much time your technician might have to do that, whether they can pre-prepare the discs to completely remove that hazard from the experiment. And then thinking about our glass equipment, if that breaks, it will smash and it could cut the skin. So to reduce the likelihood, make sure that the test tubes are always in the test tube rack. We are also creating a water bath, so there is a possibility that you might be using hot water that could cause scalds, so students must handle with care. Now, the last thing to point out, this isn't a risk, but obviously we are looking at the stain, the pigment, the betulin pigment inside of beetroot. It does stain, so I would highly recommend students should be wearing their lab coats, and if there are gloves available, they might want to wear gloves so they don't stain their skin. So let's go through the method then, starting with I'm going to make the water bath. So I'm actually just using hot water from the tap because most taps, the water is about 40 to 45 degrees C when it comes out of the hot tap. And I'm going for a 37 degrees water bath, which actually that is spot on 37 at the moment. The temperature is going to drop though, so I do have a kettle so I can add hot water throughout to make sure it stays at 37 degrees C. And make sure your students are aware to overfill the water bath, because as soon as you put your test tubes in, the water rises and it could overflow. So that's my water bath made. The next thing then is I'm going to label my test tubes. And I'm actually going to double up and have a test tube for each concentration twice. And I'll point out why as we go through. That is my 0. I'll put that in the water bath for now. So 0%. This will be my 20%, 40, 60, 80, 100. I'm just going to label again on these spares. So next then I'm going to add 5 millilitres, which is the same as 5 centimetres cubed, of each of these pre-prepared concentrations to the correctly labelled test tube. So I've got my distilled water here. It would actually be easy if you had another beaker, you could pour that into it, because you might not have a syringe that can fit all the way down. One of our control variables was the volume of the solution, so students do need to make sure they are accurately measuring this. So make sure that they know that the bottom of the plunger should be in line with the value. So I can see 5 just here. That is where we should be going to. And this was distilled water, which means 0% ethanol. So that goes to my 0. And I'm going to put that over there so I don't use it again. We go to this one next, which is 40%. 60%. We've got 80% here. 20%. And then lastly, we've got the pure ethanol. So I'm just going to pour it into here so I can get my syringe in for the 100%. So we've got all of those solutions in there now. I'm actually going to put a bung in all of them and let them reach the 37 degrees C. So that's now going to take a few minutes just to reach the same temperature. And we need to make sure that we do wait about 5 minutes to do that and then test, because we want to know that the solvents are actually at the temperature that we think the water bath is at. So at this point, I'm just going to go through the why then I've put the bungs on top and why these were all covered in cling film, because this is a key question that students get asked in the exam to make them aware of. We have to put the bungs on top and in this case cover it with cling film because the water within those solutions will evaporate. And if the water is evaporating, that now means we'll have a more concentrated solution remaining. So what we thought originally was, let's say we've got here 20%. If water is evaporating out of that throughout the whole experiment, we now don't actually have 20%, it might be 25% instead. So that's why we keep bungs on. It's so that no water can evaporate, so that the concentration inside there is the concentration the students are expecting that they have. Now I'm going to put on some gloves before the beetroot comes out. Even though I'm going to be using forceps, I'm still going to put gloves on just in case so I don't get stained red. And we've got a selection of beetroot discs here. So we've got 6 test tubes. Depending on how much they have will determine how many pieces you can put in. I can actually see here already though, some of these are not exactly the same size. So that's one thing students should be aware of, that they aren't necessarily all the same size and they should be. So that's one control variable. So if you do have this, try and make sure they are cutting it to the same size or picking pieces that do all look pretty much the same size. You can also measure the mass to try and help with this, but the surface area is going to impact it as well. Now, after these were cut, these were then rinsed, and that's another really common exam question. Why would you rinse and tap the outside dry before you place it in there? And that's because when the beetroot is being cut, you're going to be cutting through some of the cell membranes and releasing that pigment. So we need to wash them to remove that pigment, because we are only interested in how much pigment is coming out of the cells as an indication of the permeability of the cell membrane. So I'm now ready to put my beetroot discs in, start the stop clock and leave them for about 10 minutes. I'm going to check at 5, have a look at the results, and if I think it needs longer, we'll go on to 10, and then I'll recommend to you which time worked best. Let's also just double check. We're about 33, so I'm going to add a splash more hot water, and I'm going to go for 3 discs each. If they're working in pairs, that'll be better so they can do this quicker, because otherwise the timings are going to be slightly off for some of them, in the sense that's the ones that go first. Oops, lost a bit there. Will have more time than others. Mm. 100, there we go. So that's one thing just to be aware of. I've noticed that because ethanol is a solvent, some of it must have got on the outside because it's actually washed off this here. But by default or process elimination, managed to work out that was my 40%. So you might want them to label the bottle, the test tube, and then also the bung on top as well. Okay, we'll leave that going. So I've left this now for 5 minutes. I now need to put my samples into the cuvette. Just to hold them up to have a little look, we can see, so this one here is 0%, we've got 10%, we can see there are visible differences here. What I'm going to do is just invert each one to make sure that it does mix. We've got all that pigment into the solvent. And for this one, this was my 40. I'm just going to pour that into here, just so I don't have the beetroot submerged in it anymore. This one's 80%. So we can see that there is a difference, but if we were going to just do this based on our qualitative descriptions, it's going to be really tricky because these 4, they do look pretty similar. We can see that one's noticeably paler, and then this one's the next darkest, but this is why using our colorimeter is going to be best for this. So now we've got all of those results, let's use the colorimeter. And a few things about the colorimeter to start with. Students, because we're using red liquid, they should set the filter to green because you want the opposite color on the color wheel. We also need to add the liquid to the cuvettes, and with the cuvettes, they need to make sure that they're filling it up about 3/4 full, so that it's full enough that the light will be shining through the liquid, but it's not so full that they could spill it into the colorimeter. They also need to make sure that they're holding it at the edge edges so that they're not going to put their fingers on the part where the light shines through. On this one, the edges are indicated by these ribbed sides, these ridges, and you've got a frosted triangle on the front, so which bit should be facing forwards. But they don't want to put their fingers on the part where the light shines through because the grease from the fingers could affect the absorbance of the light. So I'm now going to pour in the liquids into these and record my results. Now, before we test it, we do have to standardize our measurements so that we know that every time we're saying 0 is whatever the absorbance is with distilled water. So I've got 1 cuvette that I've filled up with just distilled water. Place it in until you feel it click. And then I'm going to press R, which is our reset, which basically means we're standardizing to say that is 0, and that's what we're going to compare to. So now I'm going to pour in my 0, making sure I'm only touching on the edges. Put it in so it clicks. T for test. And my absorbance is 0.34. Next then 20%. 0.63, 40%, 2, which I think is actually the maximum. So I'm just going to 0 again, just to make sure. Oh, hang on. 0 again, pressing the R. And then test. So it is still saying 2. I suspect these are all just going to say 2 now because 2 is the maximum. And we'll talk about what you could do to overcome this. So the 4 samples that I said looked to the eye to be the same colour did actually come out as the same absorbance, but I do think this one is the darkest if we have a look at it, but it's capped or it's reached that maximum that it can get to, which is 2. So the final thing is making sure students are tidying all away safely. So their liquids will all need to be poured into the organic waste. And when I say liquids, that is their ethanol solutions. And anywhere they've still got the beetroot discs in, you might want to have a waste bucket like this with a sieve over the top. So they can do that, collect all of the beetroot at the end, and then any liquid in there can also be poured into our organic waste bottle. So essentially what this means is I've got to the point where there's so much pigment in it, it can't differentiate between the rest because all of them are at maximum absorbance. So different ways to overcome this. I actually put 3 discs of beetroot in. So you might want to try it with only 1 disc of beetroot in. I left it for, in the end, I think it was actually about 7 minutes before we got to the point of pouring it all out. So you might want to do 1 disc of beetroot, 5 minutes. And what that will mean is you'll get less pigment coming out, but it might mean then that you don't hit that top amount and you will get that full range of results. So once the students have that data, if they have time to do repeats, even better, because then they can have their multiple trials, calculate a mean, and then they can plot the mean absorbance. They would need to plot their absorbance against the percentage concentration of ethanol, and then they can plot their calibration curve. And from that, if they then were to test unknown concentrations of ethanol, place their results on the colorimeter, they can read off on the graph what concentration of ethanol they have based on that absorbance, like we can see on this graph here. So finally, just a few top tips to help your students succeed in this practical. Number 1 is making sure you're using fresh beetroot, not pre-packaged, even if it still says it's raw, because you get better results. Number 2 is making sure that the discs are cut to even sizes so they are the same size and surface area. Number 3, making sure that the beetroot is rinsed and then patted dry after cutting. Number 4, don't let the beetroot dry out before you then do the experiment. So you do need to prepare the discs and then use them straight away. And number 5, making sure that you do shake them consistently like we saw at the end. So you do have that equal distribution of the pigments. Now, common mistakes is basically not doing those things. So not cutting to the same size, not checking your water bath to make sure it is at the correct temperature, not increasing the temperature if you need to. And then actually, I'd say a mistake that we saw here in me doing it was using too many discs or leaving for too long so that you actually get so much pigment coming out that you can't distinguish between the absorption. So make sure you do trial this beforehand and maybe trial it with 1 disc, 4 or 5 minutes. But that is it for this experiment. Don't forget you can get the complete free guide in the description below, and make sure you're subscribed so you don't miss out on any of the next videos. Welcome back to another video going through the required practicals for A-Level Biology, and in this one we are doing a heart dissection. This links to AQA required practical 5, but other exam boards also have a heart dissection. You don't actually have to do a heart dissection for the required practical. It's any organ that is linked to mass transport or gas exchange for plants or animals. So I'm doing a heart dissection. It could be the fish gill practical instead, it could be lungs, or you could use plants. Everything that I'm going to be going through today, you can download a free copy of my equipment, method, risk assessment, and the idea of the results. So click the link in the description if you want to get that for free. And make sure you subscribe so you don't miss my entire A-Level Biology required practical series. So if you are about to do required practical 5, a heart dissection, I'm going to be going through with you what you can show on the outside, how to do the dissection, key things that students should be aware of, common mistakes, tips, tricks, as well as all the essential information like the risk assessment as well. And a big thank you to Woodford County High School for letting me film here today. So for the students, one of the key focuses of this practical when it gets to the exams is knowing how to work safely, how to do scientific drawings of their results, and being able to identify the structures that they actually learn in lesson. So as we go through, those are some of the key things that I'll be pointing out for you. The key equipment that you're going to need for this practical is a heart. Now we typically use a sheep heart. It is similar in size to the human heart, and you can tell this by if you put your hand as a fist. The size of a fist is approximately the size of your heart. That's something I'd always show the students as well. We've also got a sharp scalpel. We've got sharp dissection scissors. We've got our forceps, and we've also got a mounted needle. As well as that, I've got a beaker of disinfectant. We've got Virkon, but you just need to have a disinfectant that is accepted by CLEEPS. And I've got my dissection board, and we actually use these absorbent pads to help absorb any of the liquid and blood that might come out of these. All of that needs to be tidied away in a separate biohazard bag at the end of the practical, as well as the heart and any gloves that are used. Before any heart dissection, I always show the students the key structures that they need to know on the heart, on a model, and then actually on the outside of the heart as well. So let's take a look. So here we have the model of the heart, and the key things that I like to be able to identify on the heart with the students are coronary arteries that run on the outside of the heart. Also, the atria are usually quite obvious. Sometimes they get cut off though, so you can't always see them. And then we're going to look out for as well the aorta and the pulmonary artery. It's unlikely that we'll have the vena cava and the pulmonary vein in shock because they often get cut off when we get our hearts. So if we have a look, we can see first of all on the outside, I'd point out the difference between the dark tissue here is the cardiac muscle, and the white that is actually fat on the outside of the heart. But that makes it easier to see the coronary arteries, where we can see them running along these line-like structures, and those are the blood vessels that provide the cardiac muscle with oxygenated blood so that they can respire aerobically so the heart can contract and relax. And it's if one of those get blocked, that's when you don't get the oxygenated blood to the heart, muscles can't contract and relax, and you can have a heart attack. You can actually see the atria, which are these small chambers at the top. So small that often students don't realise that those are the atria themselves. So it's worth pointing that out to the students. We've got one at the top here, we've got our left and right, we've got the other one here. So those are the atria. The ventricles are the chambers here, and when we cut into those, we'll be able to see the thickness of the cardiac wall of the chambers of the ventricles. Now the blood vessels, I have got 2 that are still intact here that we can see. We've got here the aorta, and we've got the pulmonary artery. We can tell that these 2 are the arteries because they've got much thicker walls. So they've got a thicker muscular layer in the wall as well as that thicker elastic layer. And we can tell that this one's the aorta because it is much thicker compared to this one over here, which would be the pulmonary artery, which is taking blood away from the heart to the lungs, whereas the aorta is taking the blood away from the heart to the rest of the body. The vena cava would be the vein that's going into the right atrium, and the pulmonary vein would be the one that's going into the left atrium. But we actually can't see these because we can see there's actually been an incision already into the atria for the heart at this point, which is often the case. And I can see here, I've got an incision here where, depending where you get your hearts from, that is often the case. So just make the students aware. So that's the outside of the heart. Here are the key structures your students must know on the outside of a heart before they do the dissection. I'm about to dissect this sheep heart. Watch along for all the key information that you need to know for your A-Level Biology students. After the students have had a look at the outside of the heart, they would have already identified the atria, coronary arteries. I'd get them to take a photo of that because they might need that for their scientific drawings, as well as taking a photo from this angle so they can see the blood vessels as well. After that, we can now see here are the atria, and in mine, they've already been cut into. So the key things that I'd be pointing out to the students here is, this is your atria, that is the cardiac muscle. You can see just how thin the cardiac muscle is in the atrial wall. And that's because they don't contract with much force, because they only have to pump the blood down into the ventricles, which is not a far distance. Once they've identified those, they can then take photos of that as well. We're then going to cut in so we can see the difference in the thickness of the ventricle cardiac walls, and so we can see some of the valves. Be warned if you are squeamish. One advantage of them actually already cutting into the atria, and actually you can cut into the atria to do this, is the way that I then check I'm going to be cutting in the right place is you can get your finger into the atria, and that then leads directly down into the ventricle. So my finger is now down here in the ventricle. So I know that if I cut where I've got my finger, I will take my finger out, but where you cut there, you know that's going to go through the atria and into the ventricles. We'll then be able to open it and see at that point. So that would be what I would do next. Now, my actual preference is to use scissors. In the exam, they'd want you to say you use a sharp scalpel cutting away from you. I actually find that if you've got sharp dissection scissors, it is far easier to control and actually easier to cut. So I'd be cutting through. We've now cut in so we can see here was the atria. This is the atrioventricular valve. We imagine when it was all round like this, it was all connected. So this is the valve that prevents the backflow of blood from the ventricles when they contract up into the atria. And you can see it's a really thin layer of tissue. And I do show the students that it is really thin, but you can see it is tough. I'm pulling on that quite hard. And the tendons here are connecting the atrioventricular valve to the ventricle wall. So you can see that valve really nicely there. I'd get the students to hold it out. You can use your mounted needle to pin it down. I'd get them to take a photo at that point so they can see that structure really, really clearly. The next thing is looking at and pointing out to the students just how thick that wall is. So this is your ventricular wall, or the wall of the ventricles, made up of cardiac muscle. And we can see that's a really thick layer, especially if we go back and have a look at the atria where it was this really thin layer in comparison. So again, I'd get them to take a photo, because when it comes to doing their scientific drawing, these are the sorts of things they'd need to be able to label. That this is the ventricle, that this is an atrioventricular valve, we've got a heart tendon, we've got the atria, and I'd be getting them to label, or they would need to know, that this is the thick muscle of the cardiac wall of the ventricle. So now, same thing again, I've put my finger through the atrium, so I know that that's definitely going to go down into the ventricle, and I'm going to cut through this bit, and I think this is the left side, judging by how thick it is, so this must be the right. So let's have a look just to confirm. Yeah, so we can see, again, we've gone through our atria, which I've actually completely cut now. That was, if we put it again round like that, that was the atrium. We can see the valve with those tendons. And now we can see the thickness of that ventricular wall. This is the right side of the heart. And we can see just how much thinner it is on the right compared to the left side of the heart. And the students can annotate this on their diagram. They need to know that the left side of the heart, the ventricle has got thicker muscle in the cardiac wall, because that is pumping with more force. It contracts with more force, pumps the blood out of the aorta at high pressure, so it can reach all of the body. Whereas the right ventricle, we've got thinner muscular wall. That's because it doesn't need to contract with as much force, because it's pumping the blood from the right ventricle through the pulmonary artery to the lungs, which lie directly behind the heart, so it's not as far a distance. And also, we don't want the blood going into the capillaries in the lungs at high pressure, because that high pressure could actually damage the capillaries, and we want it low pressure, so it flows through slower, so there's more time for gas exchange. So now I've actually cut down both ventricles. We can see this section in the middle of the heart, that is dividing the right and the left side of the heart, and that is known as the septum. So that is what is separating the two sides. I'm actually going to cut down the septum now, so we can then see at the same time the right side and the left side to see the difference in the thickness. I would get them to take any photos though before they do this, because you can't undo it. So there we go. I've now cut the heart in half, and we can see this section here. That is the septum separating the left side of the heart and the right side of the heart. So we now know that these are our two ventricles. We've got the two atria at the top. We've got the atrioventricular valves. In this one, we can't actually see the semilunar valves, but those are the valves that are between the two arteries that leave the ventricles and the ventricle, but they're not actually visible on this one. So I'd then get them to take a photo of this, and then they can label that. We've got the left side of the heart. So this one, that is the left, that's the left. You can tell it's got that thicker muscular wall. We've got the right side of the heart. We've got the septum. This is the apex, meaning the very tip of the heart. These were the atria at the top. We've got the atrioventricular valves, the tendons connecting to those, and on this section we can still see the aorta and the pulmonary artery. And again, the final way we can check we've definitely got those blood vessels the correct way, as well as the thickness. We've said that this is the left side of the heart because it's got that thicker muscle. And you can actually put your finger up to go through that blood vessel, and we can see from the left side, the left ventricle is going through this tube, which would be the aorta. From the right side, your finger goes up through that one, which would be the pulmonary artery. One of the key ways the students have to present their results with this is with a scientific drawing. So here are the key things that they need to know. Number one, there has to be a title of what they are drawing. Number two is knowing that you shouldn't have any colouring, any shading at all. So I've got a picture that I've taken, then I'd be doing my scientific drawing matching that. But I'm going to do a demonstration here rather than having it literally match picture. So on this one, we can see that we have just got the outlines. There's no colouring, there's no shading. There are things I've deliberately left in which are errors that you can point out to your students. So we can see here that we've got two lines that don't connect. So there's a gap and there's an overhang. Same here, we've got an overhang and we've got a gap between those. There shouldn't be any gaps, there shouldn't be any overhangs. So the student would have to redraw that bit. This is why we always do it in pencil and a sharp pencil. So they have really good precision in their diagram with no overhangs as well. Next, they would need to indicate the scale. Now, obviously, I've done this on a whiteboard, so it is much, much bigger than the actual size of the heart. But if they're doing this into their lab book, which is probably A4, I'd recommend that they attempt drawing it to scale, and then they can just write to scale on their diagram. For me, this is probably at least 10 to 15 times bigger, but I'd have to measure to work that out. But they need to say the idea of the scale. Next then, they need to label and annotate their diagram, making sure that none of their label lines overlap with each other. So this one here, and they should do this with a ruler, we could say that this is, so this would be our right ventricle wall, and in other words, I'd also say cardiac muscle. On this side, we could then have, on this side, we've then got the left ventricle wall, and the diagram should visibly show that you've got a thicker cardiac muscle wall on the left compared to the right. Now, I've actually drawn this using the heart that I had, but I've actually drawn the one that I had a better image of. But it is worth noting that normally when you draw the heart, you're imagining it is the heart against you. So you always have the left on this side, the right on that side. So that is something to note to the students, that what they might be drawing is what they had a better image of and dissection of. But bear in mind, in the actual exam, you always have the left on this side, the right on that side. Then they can add the other labels, such as the atrioventricular valves. We've got the heart tendons, we've got the septum. We could label this as the apex. We have got our two different blood vessels at the top, and for us, we put the finger through and we worked out that was the aorta, that was the pulmonary artery. These are our two atria. So we've got the left atria, the right atria, and then when it comes to annotations, this is where they could start to explain why you have the thicker cardiac muscle in the left ventricle compared to the right ventricle. We could label the role of the tendons, the atrioventricular valves, and how the septum is separating the oxygenated from the deoxygenated blood as well. So once you've then done all of the dissection, you've got all the photos that you need. Now we need to make sure that we're sticking to the risk assessment and tidying up. So anything that has touched the heart that would go in a bin needs to go into a separate bin. All of the equipment needs to go into our disinfectant. And then once I've put all of this into a separate bin, I take my gloves off inside out, put that in the bin as well, wash my hands with soap, soap, and then make sure I disinfect the entire table. Hopefully you found this video helpful, so you now know what to point out to your students, what are the key things that come up linked to the risk assessment, the structures they need to know how to identify, and how to actually do the experiment to show the students in advance or for them to use as part of their prep. Don't forget you can download for free the plan for this practical, and make sure you subscribe so you don't miss out on any of the next videos. Hey everyone, and welcome to Miss Estruch Teach and Tell, and in this video I'm going to be going through the required practical 6, the aseptic technique to create an inhibition zone for A-Level Biology. I have got these videos for all of the required practicals, so if you aren't already subscribed, click the subscribe button now so you don't miss out on any of these. Plus, for all of the practicals, you can download for free the entire guide for each practical, which includes the aim, the method, equipment, risk assessment, key mistakes, and key tips to explain to your students as well. And a big thank you to Woodford County High School for having me here today to film the practicals. So in this experiment, we are going to be testing a range of different antibiotics with E. coli to work out which antibiotic is the most effective at killing this strain of E. coli. And within that, the students are being assessed on their aseptic technique as well. In this experiment, we're going to need a Bunsen burner to help keep our aseptic conditions. You're going to need matches to light the Bunsen burner. The antibiotics that you're going to be using to test with. I've got sterile spreaders here, pre-sterilized. I've then got my E. coli bacteria, which we can see here, the strain K12, which is a safe strain to use in schools. I also have a sterile syringe to use to transfer the bacteria onto my plates. I've got my pre-sterilized forceps to move the antibiotic disc onto the plates. Permanent marker to label the Petri dishes, tape to tape down the lid of the Petri dishes. I've got pre-poured agar plates, which are all sterile. So we've got sterile agar plates pre-made. And then I have my Virkon disinfectant, which I'm going to be using for the equipment after it's touched the bacteria. And I'll also use this to sterilize the table at the start and the end. And then finally, I've got some cotton wool to use to dip into my disinfectant to clean down the surface at the start and the end. So step 1, I need to wash my hands with soap to make sure that I'm removing any microbes on my hands so it doesn't contaminate our plates. Next, I'm going to dip my cotton wool in the Virkon and just sterilize the section of the workbench that I'm going to be working on. It's now at this point that I would put the Bunsen burner on. I don't actually need it on the roaring flame yet, so for safety reasons, I'm going to leave it on the yellow flame so we can see it, but just have it on ready so that I'm minimizing the amount of things that I'm touching after I've already washed my hands. So one of the key things students need to know are the aseptic techniques, and that's the common thing that comes up in the exam. So what they need to know is, first of all, they must wash their hands with soap, or you could also say, wash the surface with disinfectant at the start and the end. And that goes for the washing your hands with soap. The mark scheme is really particular. You have to say with disinfectant, you have to say with soap. The reason for those are you need the soap or the disinfectant to actually kill and remove other microbes that could contaminate your plate. And in terms of at the end, it's to kill the bacteria you've been growing so you don't then spread it across the school. The other things linked to the aseptic technique, we briefly spoke about when we went through the equipment, but any piece of equipment that's going to be used has to be sterile, meaning completely clean with no other microbes on. So we've got pre-sterilized forceps. These were sterilized in an autoclave, so students need to know that. They could be asked how you could sterilize other equipment. You could either sterilize it in steam, an autoclave, or in the Bunsen burner. But if you do have something that is plastic, then that can't go in the Bunsen burner because it would melt. One of the other key things for the aseptic technique is before you use your E. coli, you would need to flame the neck of the bottle through your Bunsen burner in the roaring flame so it's hot enough to kill any microbes that might have been on the top. When the students are working, they also need to make sure they're working near the Bunsen burner because this is so hot, you're going to get a convection current drawing the air up into the flame, and then as the air cools, it sinks again. So you constantly have this circulation of sterilized air around where you're working. When working with the agar plate, we need to make sure that we're only ever opening this very, very slightly slightly at an angle so that we can get into work, but we're reducing the amount of air that could potentially land on the dish that might contain microbes on it. And then at the end, we're going to see that any piece of equipment I have used is going to go into the Virkon, so it disinfects the equipment to kill any microbes that might be on that. So the key risk assessment for the students is mainly what we've gone through with the aseptic technique. So washing your hands with soap, disinfecting the surfaces, because that is now going to be protecting you and other students from transferring any microbes out of the classroom. So they must make sure they wash their hands with soap at the end. Because we're working with a Bunsen burner, we have to be very aware that anyone with longer hair, the hair's tied up, and you don't have any loose things that could, as you lean over, dangle into the flame. Also wearing goggles just to protect my eyes, just in case we get any splashes of E. coli. And then the big one, we are using a safe strain of E. coli. It's non-pathogenic, but also if a student does spill any E. coli, they must make a teacher aware immediately, because we'll need to soak that area with disinfectant and the teacher will need to clean it up safely. When we come to incubate our plates, we need to make sure we put tape only on the two sides so that air can get in, so we have oxygen, aerobic conditions, so we don't accidentally grow any harmful anaerobic strains. And also the plates must be incubated at 25 degrees C, so it's warm enough for bacterial growth, but not so warm that we encourage excessive growth and potentially get large population sizes of pathogenic bacteria. But now let's get into the actual practical, and as we go through, I'll point out some key mistakes students make so that you can point it out to them so they don't, and some key tips as well. So I've washed my hands with soap, I've disinfected the surface, I've got my roaring Bunsen burner flame set up. I've actually got some labels already on my Petri dishes because these were pre-made by the technician. So at the moment it's just saying it contains only nutrient agar, and that was created on this date. Because I'm now going to be adding in something else, you have to add in what is going into the plate, and we do it around the edge so it's not going to block our results at the end. I'm going to put my initials as well, so the students should put their initials because in the class they need to be able to identify which are theirs. And I'm adding E. coli. So that is now my new label that I'm adding to the bottom, at the corner, at the bottom. So if the lid does fall off, we still know what is on the actual agar, and right at the edges so it's not blocking your plate and your results at the end. So now I'm ready to flip it over. So I'm now ready to get my bacteria from my bottle into my plate, and I'm then going to spread it evenly across the bottom. So the key things to be aware of here is we've got our sterile syringe, sterile spreader. One common thing that students often do wrong at this stage is they open up all of the equipment, put it on the table, and then they leave it. That gives opportunity for microbes to land on it and it to become contaminated again. So we do not open our items until we are literally ready to use them to reduce the likelihood of contamination. Another key mistake that we'll see, and I'll point this out as I go through it, is when using the spreader, sometimes students don't actually spread all the way across their plate to the edges. So you then don't get the complete bacterial lawn covering the whole dish, and that can impact the ability of you being able to see the inhibition zones. So let's have a look at this. First of all then, I would flame the neck. Now I'm going to open up and get my syringe out, and we actually use 0.7 mil when we do this. We found that that's enough to get good results, that we can actually see the growth and not so much that it's really wet and splashes around on the plate. I then put the lid on straight away so I can't accidentally knock over the bacteria. Working near the Bunsen burner, so we've got that convection current. Then it's going to open it, only slightly at an angle, put that bacteria onto the plate. I'm going to close it now, and before I spread it, put the syringe into the Virkon, but get your students to actually pull the syringe up so it draws the Virkon inside, so it's disinfecting the inside and the outside of your syringe. Next, I need to open these spreaders. We can see it was a sealed sterile pack, so as soon as this is opened, they're no longer going to remain definitely sterile. So again, you want to only slightly open the lid, and you want to be working near your Bunsen burner. You need to make sure you're spreading it all over plate. And it's hard for students to do this because it is all colourless, so you can't actually see for definite, and you'd want to try and not add in microbes. So I tend to just go around it almost like a clock face, just to make sure we're definitely getting all the way to the edge of the agar plate. And then immediately the spreader is going into the Virkon. That's the last step, just putting the antibiotic disc on. Final step then is we need to put the antibiotic discs onto our plate. And this is where we're going to use our sterile forceps. Now you might not have an entire ring like I have here. They might just have individual pieces that they pick up and place on. But I've got the entire ring. Now these have been sterilized already. As soon as we take the top off though, they're now exposed and they might not be sterile. I would still actually put them through there just to be sure that they are definitely sterile. But because I've now put them in the Bunsen burner, I do just need to leave it a little bit to cool down so it's not going to kill any microbes if it does touch that. Now this has actually got a little bit here on the mast ring. That's what we call this ring of antibiotics that I can hold onto with the forceps. And this is where we get a common mistake students make, that once they put it down, you need to put it down correctly the first time because the antibiotics will imprint straight away and it affects your inhibition zone. And you don't want it too close to the edge so that you can't get a circle. That is the inhibition zone. So I need to lift this up and make sure I place it right in the middle, which is the tricky bit. And then those forceps go into our disinfectant. Final step then is we will tape our lid down so it can't come off, but you need to make sure you are only putting a piece of tape on either side. Just like this. And that's enough to keep the lid on. But if we put tape all the way around, then oxygen can't get in. And that is then when you might grow anaerobic bacteria. And then when you're ready, you would place your dish upside down because at the moment, the bacteria that are there, they're going to respire, and water in the form of water vapor is one of the products of aerobic respiration. And that water vapor is then going to rise to the lid, condense, and it can drip back down onto the dish. And if you then have pools of water, that can affect the inhibition zones. So instead, we flip our dish upside down so any condensation lands on the lid. Now, sometimes I do get the students to wait just about a minute or so before they do that because it is still quite wet in there and everything might just fall upside down. But let's give it a go. Okay, so we've left it long enough. It is now all staying on the top. The dish has stayed there as well. And that is our plate ready made to now go to be incubated at 25 degrees C for about 48 hours minimum and about 1 week maximum. Otherwise you'll have too much growth and it could be potentially harmful. So final step, I'm going to turn off the Bunsen burner and then we need to clean the surface with disinfectant and wash our hands with soap again. So there we go, just clean down the area that I worked. And I'll then be washing my hands with soap also. It then does come to the results. The students will have their plates back, and I actually often find it's useful if they do use a pen to draw around the inhibition zone on the actual Petri dish, so it's easier to take a photo and visibly see, and then accurately measure the diameter of the inhibition zone with a ruler in millimeters. Once they've got the diameter of all of their inhibition zones, they can then put their results in a table, but I recommend that you do pull the class data, so then they have their repeats, they can calculate a mean, 2 times the standard deviations on those bars, and then they can actually compare to see are there any overlaps in the standard deviations to see do they have any significant differences between the means and therefore how effective each antibiotic is at killing that particular strain of bacteria. You could also get them to calculate the student t-test, which would be another way to see do you have a significant difference between the means and therefore is it a significant difference. Now so far I've actually just mentioned doing it with the diameter. It would be better if you had it as the entire area. So once they've measured the diameter, they can then do pi r squared to calculate the area of each inhibition zone and pull their data for the mean area. So that's the data they're going to be collecting. I would always encourage them to take photos so that you've got that to look back on and to put in with your results. And also it's useful to be able to see, did they work completely aseptically? And we can tell that by, do they have any contaminations on their plates at the end, which would look like different colored dots, often like a bright orange color or a darker yellow color, indicating different bacteria has grown other than the E. coli. Or if you have anything really furry, then that is your fungus that is growing. So that is it for Required Practical 6. If you have found it helpful, don't forget to subscribe because I go through all of the required practicals and you can download for free the complete guide with all of my tips and tricks to help you deliver this as smoothly as possible. In this video, I'm going to go through with you the required practical for chromatography, Required Practical 7, A-Level Biology, and I'm going to be doing it with TLC paper. So I'm going going to go through with you the equipment, the method, key mistakes students might make for you to look for, but also emphasizing what are the common exam questions that come up linked to this, so you can make your students aware of that as well. And if you want all of that bundled together in a free guide, that is linked in the description for you. Don't forget to hit the subscribe button so you don't miss out on any of these required practical videos. And a big thank you to Woodford County High School for letting me film all of these videos here. So the aim of this experiment is for the students to be able to separate out the pigments and to know that the reason that they separate out is because they have different solubilities. And depending on the solvent that you have, you'll get a different number of pigments because the pigments may be more or less soluble in certain solvents. So they're going to then create their chromatogram, calculate an RF value, and then they can use that to compare different leaves, or they can use it to identify the pigment that they have. I'm going to be comparing two different leaves. Whatever you have available, you can choose to compare. You might want to do a shade tolerant versus a plant that isn't tolerant to shade to see if there's any difference in the type or proportion of those pigments. So the equipment then that we have for this experiment is I've got my TLC paper, I've got a ruler with millimeter measurements and a pencil. I've got capillary tubes just here. I have my organic waste bottle to dispose of the solvent safely at the end. Forceps, hairdryer to help dry the pigment if we need that. And then I've got in our tubes here, this was the spinach which has been mixed with the solvent already to get the pigments out of the leaf. I've got my coleus here, which is another leaf, which again has been mixed with the solvent. And for that one, the solvent that was used to extract the pigments was ethoxyethene, and that was mixed with propanone in a 1-to-1 ratio. Then I've got 2 McCartney bottles. Both of these are the running solvents inside that I'm going to put the TLC paper inside of. And the solvent that I've got there is cyclohexane to ethoxyethane and to propanone, and that's in a ratio of 5:3:2. The main thing here of the risk assessment is the solvents. So these solvents are volatile, they're flammable, and they give off really, really strong fumes, and it will cause severe headaches and potentially dizziness. So you either need to be working inside of a fume cupboard, or we've got all of the windows open here to reduce the chances of headaches and any issues linked to the fumes. So now we're ready to get on with our method, and I'm actually going to run 2 TLC plates, 1 for each pigment. TLC paper is expensive, so you can make this cheaper by cutting these in half lengthways, because that is still wide enough for you to be able to get a good chromatogram. And what I then need to do is draw my origin line in pencil, and then I'm going to start adding the pigment. But I need to work out at what distance up to draw my solvent front. So what I'll need to do is measure how far from the bottom up the solvent is, take that bit of glass distance away from that to know that is how deep in terms of in the bottle the solvent is, and that is where I'll draw my origin line. So it looks like 6 millimeters, and that is 18 millimeters. So 18 minus 6 would be 12. So they just need to dip it in, keep your fingers over the top to create a bit of suction, dip it on the paper, but you need to wait until it is dry before you then add the next drop, which is why I've got the hairdryer here, so you can see when it is then dry. And we'll keep going until we've done 10 drops, or at least 5 millimeters in diameter. They probably won't need to dip it again, this one. And wherever possible, closing the lids, because that's going to be letting out the solvent. So I've now got my 2 TLC papers ready to go into my 2 separate bottles of solvent. I've got my origin line drawn. I've got my pigment above where the solvent is going to reach. And then I've got my pencil and rule at the ready to take it out afterwards. So we need to make sure that we are lowering it in vertically. And I can see that the solvent was below the pigment line, so that's good. Right, so the one on the left is almost halfway done already. The one on the right, my pigment has gone slightly below the solvent line, so it might be an issue, although both of them now, it looks like a green one starting to emerge from the top. Actually, this one, yeah, you can start to see a couple of coming off the top. So I'm going to take mine out now before it reaches the top. I'm going to do it one at a time, so I can draw that solvent front on straight away. Now if we just zoom in and have a look at this one before I get the next one out. So I'd still work out the RF values for those and then look at which pigments would move or have that RF value in that particular solvent. So that one was my spinach. So I'll do the same and take out the next one. Drawing on that solvent front straight away before it evaporates. Now to get the students to take a photo at this point before any of the pigments fade. And then what I can see is we've got a faint yellow one here, and then I've got a dark grey, green, a yellow. There's 3 distinct other ones as well that I would measure the RF values for. So once the students have then completed their experiment and they've got their chromatogram, take a photo immediately, because the solvent front will disappear as the solvent evaporates, but also the pigment colour can start to fade over time. So they can always go back to the picture, plus it's nice to stick that in their lab book. What their aim is though, is to be able to identify the different pigments and try and work out which pigment they have based on the calculated RF value, and then they can research for the solvent that you used, for the RF value, which pigment is that, especially if they've got the colour as well to help back that up too. So some of the key things to point out to them: if they've got pigment spots where it is spread out, they should be measuring from the middle, the centre each time, so that is an average, and also they're standardising how they're measuring it. So they need to record from the pigment point on the origin line to the middle of each pigment, what is the distance that the pigment moved, and it'd be best to do that in millimetres to be more precise. They should also be measuring the solvent front, so that is where the solvent was initially, where the solvent ended, and that is what they're going to be dividing by each time. So it's the key things to make your students aware of then. They should always be drawing in pencil, because if they use ink, the ink is going to be soluble and that will move up as well with the pigments, and that is a common exam question for 2 marks. They should also make sure that when they place their TLC paper in the solvent, that the solvent lies below where the pigment is. Otherwise it could splash onto it, dissolve the pigment, and then you lose your pigment spots. Again, that's a common exam question. They need to make sure that their TLC paper is straight and not at an angle, because otherwise as the pigments move up, they might go off the edge of the paper. Another common exam question is that one. They have to keep a close eye, because when the solvent does reach just before you hit the top of the paper, that's when they take it out, so that you don't get all of the pigments reaching that same end point and you don't have them separated. As soon as the students have seen that the solvent has reached just before the top, take it out and they need to draw a pencil line on immediately. That's That's because the solvent's going to evaporate really quickly, and as soon as it's evaporated, they won't know the point that it went up to, and then they can't measure the solvent front. They should also make sure that they're not putting their fingers on the TLC paper at all, because that can interfere with the ability for the pigments to move up and affect the results. So that is it for required practical 7, and don't forget you can get the entire guide for this practical for free, linked in the description, and hit the subscribe button so you don't miss out on any of my future videos. In this video, I'm going to be going through the photosynthesis required practical for AQA A-Level Biology, the one with DC-PIP and looking at the enzyme dehydrogenase. We're going to be going through the equipment, the method, top tips for success, and how you can help your students understand what is going on in all of those test tubes. And if you want all of this information for free in a guide, I'll link it in the description below. Don't forget to hit subscribe so you don't miss out on all of these required practical videos. And a big thank you to Woodford County High School for having me here today to do all the filming. So if we have a look then at all of the equipment that we've got for this experiment, we've got a lamp that we're going to use as our light source. Got a large beaker over here with cold water, and I'm actually going to use this ice later on in there to make the ice cold water bath. Got a beaker here of isolation medium, 5 test tubes labelled A, B, C, X and Y. I've got some foil at the back, a stop clock, a beaker to put my distilled water in, as well as my distilled water, permanent marker to label my test tubes. I've got my ammonium hydroxide, DC-PIP, and then a selection of 5 centimetre cube syringes and 1 centimetre cube syringes. I've got my chloroplast suspension pre-made on ice. So the key hazards to make your students aware for this are, number 1, if they are going to be making the chloroplast suspension themselves, that means they're going to be blending up the spinach leaves in the isolation medium. So they need to be handling that blender with care, making sure the lid's definitely on. Most of them have a safety feature anyway that they would turn off if the lid was off, but just to make them aware of that. Next then, the ammonium hydroxide is an irritant. So we've got our warning here that it causes skin irritation and serious eye irritation. So if they do get any on their hands, wash their hands immediately, and goggles have to be worn throughout. DC-PIP is low toxicity, but if it's ingested, it could be harmful. Pointing that out to the students, but also making sure that they are wearing their goggles throughout. We are using a bright light, and depending on whether you've got an energy efficient light bulb, you can find that these metal bits on the outside can get incredibly hot and cause burns. Just make sure if you can get an energy efficient light bulb. If not, make students aware of that. Do not touch it because it will be incredibly hot. And then lastly, we do have a lot of glassware, so we should be using our test tube rack when we aren't using the test tubes, so there's not that risk that they roll off the benches, smash, and students could cut themselves. So the theory behind the experiment here is our chloroplasts, they are going to be involved in the light dependent reactions, and when we have the light source on them, they'll be absorbing that light energy. That light energy is going to be involved in the photoionization of chlorophyll, and that release of electrons eventually would be picked up by the NADP in our light dependent reactions in chemiosmosis. But when we add our DC-PIP, that can pick up the electrons instead, and DC-PIP is a redox indicator that will go from this blue color when it's oxidized, and it goes colorless when it's reduced. So the quicker that this blue color disappears, that means the quicker that the light dependent reactions are happening, electrons being released, picked up by the DC-PIP, and therefore it's going colorless. We are going to have 2 control test tubes. We've got 1 test tube that's going to be our color standard comparison, so that we have a way to standardize this subjective endpoint color change. And we're also going to be looking at the impact of ammonium hydroxide. And this does link to some exam questions that have come up as linked to using ammonium hydroxide as a weed killer. Because what we should find is the test tube that we add ammonium hydroxide to, the rate of reaction is much, much slower, or it won't happen at all. And that's because it's going to be denaturing the enzymes involved in photosynthesis, but also it can interfere with that redox reaction. So I'm going to set up all of these test tubes, and as I set up each one, I'm going to talk you through what is the role of each test tube so you can then explain that to your students. So we're going to set up the different test tubes, starting with test tube A, which is one of our control tubes. So this is going to have DC-PIP, it's going to have distilled water and chloroplast solution, and we're going to wrap it in foil. So this one is going to be our control experiment. So we're setting up exactly the same as the others, except we are blocking out the light source. So we're going to add the foil, block out the light source. So this control experiment is going to be proving that if you don't have light, the reaction won't happen. Now, because as soon as I mix the chloroplast and the DC pip, the reaction can happen, I'm going to save that until right at the end, put everything else in and not put in the chloroplast until the end point. So that means for test tube A, I need to add 5 centimeters cubed of DC pip. Just to make it easier to measure out, I'm going to pour it into a beaker. This is ice cold as well. Everything here is ice cold, and we'll come back to that when we get onto this. We need 5 in every test tube apart from C. Let's put C out the way for now. So the DC pip is our redox indicator. Then distilled water, that's going in all of them apart from the last one. And then the last test tube that I didn't put the distilled water in, instead, we are adding 1 centimetre cubed or 1 milliliter of ammonium hydroxide. So these 4 all have distilled water in. This one doesn't, because instead it's going to have the same volume of ammonium hydroxide, but because we have to have the same volume in all of them, that's why these 4 had to have distilled water. And this one has got the ammonium hydroxide. And I'm just going to take out some sort of 1. Tube B isn't going to have any chloroplasts in because this is going to be our second control tube where we're proving that the reaction doesn't happen unless you have chloroplasts present. But again, because we need the same volume in all of them, these 4 are all going to have 1 centimeters cubed or 1 milliliter of this chloroplast solution. So B is just going to have the isolation medium that the chloroplasts were blended in. Prove it's nothing about the isolation medium causing the changes, and it is the chloroplast suspension. So the final thing that I'm going to be adding is 1 centimetre cubed of the chloroplast suspension to all of the test tubes apart from B. As soon as I do that though, the reactions will start. So I'm actually going to prep my tube A now. This is one that needs to be completely covered. I'll leave a little bit spare on the top where I can cover it afterwards. Once I've got my solution in. So just to go through at this stage then, before I add in the chloroplasts, what we have going on in each test tube. We said A and B are both control experiments. A, we are expecting there will be no change. It will stay this DC pip blue colour because if there's no light energy for the chlorophyll to absorb the light energy and therefore no photoionisation of chlorophyll, no electrons released, DC pip can't pick up electrons. So the reaction won't happen. Test tube B, this one here, we will have light going in, but I'm not going to add any chloroplasts to it. And if we don't have the chlorophyll present in those chloroplasts, and there's nothing there to release the electrons for the DC pip to pick up and to decolorize. Test tube C. Now this one I can actually add the chloroplast solution in now because this is going to be our color standard. There's not going to be any reactions happening here. So the point of test tube C is for us to compare all of the other test tubes to, and when they become the same color as test tube C, that indicates the end point of the reaction, because this is what the color of the chloroplast suspension is by itself. So when the DC pip is fully decolorized, it should look the same as test tube C. So that's the purpose of C, to help us standardize these qualitative endpoints of the experiment. And then lastly, X and Y, which I haven't talked about much yet. These are our 2 experimental test tubes. We still need to add the chloroplast suspension to both of them, but as soon as I do that, the reaction starts, which is why I'm holding off. X has got the chloroplasts and the DC pip. It's going to have the light source, so we're expecting this one to change color first. Y has got exactly the same, except we've added ammonium hydroxide, which is likely going to affect the enzymes involved in the reactions, so that one either won't decolorize or will take much, much longer. And the enzyme that we're referring to is that dehydrogenase, which can remove hydrogens. Hydrogens can split into protons and electrons. Last thing then before we get started is everything has been ice cold, and that's because as soon as you homogenize, so break open the cells, you're exposing enzymes to the chloroplast that they may not have previously been exposed to, and that then means those enzymes could damage the chloroplasts. So they're ice cold so that the enzymes aren't going to be working. So at this point I'm going to move that into my water bath. C, I'm going to keep on the side here because that's going to be what I'm comparing to. We get my stop clock at the ready, and then just mix up a chloroplast suspension and get my 1 ml in all the others as quickly as possible. So need that in A, X, and Y. And there we go. So into the water bath. And I'm going to apply my light source so that we have light energy for those light-dependent reactions. One consideration you could get the students to think about as well is the position of these test tubes, because I've only got a light source here on one side. So are they all receiving the same light intensity? So that's one consideration. They've got multiple lamps, they could position it. I'm going to try and position it here so they are all receiving the same amount of light energy. So while that is reacting, this could take 5 minutes. I've had it where it's gone on longer than my hour lesson. So if you are finding that it's taking ages for those colours to change, then tweak the volumes and the concentrations a bit. I've got here 5 centimetres cubed of DC pip, 1 centimetre cubed of the chloroplast solution. In the past, when it was taking too long, I decreased that to 2 centimetres cubed of DC pip, and I increased the chloroplast suspension as well. Get your technician to trial it, or if you've got the time, trial it and see what concentrations and volumes work so that you do get results in the space of time that you have for your lesson. But just some other key tips in general. So making sure students have fully covered test tube A, because that is our control where we don't want any light getting in. Make sure they don't forget about test tube C and they are using that to compare to their test tube, so they know that is the colour standard to compare to, and that they understand that is because colour change is subjective, so we need some way to standardise it. Now you might use a colorimeter with this, in which you take out samples, put it in, test the absorbance, and when the absorbance is the same as the absorbance for this test tube, that is your endpoint as a way to standardise it even more to make it quantitative. Make sure the students do start immediately with their stop clock as soon as they've got everything in, because that's when the reaction happens. And they can give it a little stir just to help make sure we do have an even mix throughout the test tube, and hopefully just to speed things up a bit. But they need to make sure they are doing it consistently across all 3 of those test tubes. So at the moment, we can see that mine's already up to 19 minutes, nearly 20, which for the length of time you have in a lesson, ideally you want it to be less time than that. So what I'd recommend is you would use a lower concentration, so dilute your DC pip, or you could add more chloroplast suspension, or you add a lower volume of the DC pip. However, I've done this so many times across the years, and different volumes and concentrations have worked each time. And I think it really depends on how fresh your spinach sample is to tell you how active your chloroplast suspension is going to be. So it's not a matter of me telling you this is the perfect volume. You will need to try it ideally in the day on that morning to see what will work for you. And we can see it is still working. The colours are changing. It's just, it can get quite boring for the students if for 20 minutes they're just sat watching this, nothing happening. But if you've got another activity, they could do that. But it'd also be better if it was quicker, so there was time for repeats. So the main thing is, if you have got the time, trial it the day you're going to do it, so you know that for the spinach sample you have, what volumes work best. Now for me, my X is nearly done. We can see those are almost the same level of green now. We can see that B hasn't changed colour at all, so that proves that if you don't have a chloroplast, the DC pip doesn't lose its colour. Let's have a look at A. So this is the one with light. So for this one, we don't have the colour change. It does look slightly green on the bottom, but you can make students aware that that is probably because some of the chloroplast has just settled to the bottom. It's not that the DC pip has changed colour. So A has proved that we do need light. B has proved to us that you do need a chloroplast suspension. C was just my colour standard to compare to. And from this, we can see that X is changing colour a lot faster than Y. It's still not completely the same colour as my colour standard, and Y is nowhere near that point yet, and we're at 20 minutes. Now you might find, I have found in the past that my Y tube never changed colour. I even left it running while the students have finished the lesson, and I said, I'll give you the data later. And it could be that the concentration of the ammonium hydroxide was so high that it did actually denature all the enzymes. The reaction didn't happen. So if that's the case, I wouldn't worry about it too much, because the students need to just have an understanding of the theory. And then in the next lesson, if you're doing the write-up, you can provide them with a set of results for tube X and tube Y, so they can then still go on to do data analysis and a statistic based on that set of data. So in terms of how they could present this data, they could have this as a bar chart where we've got time taken for reaction to occur, tube X, tube Y. You then plot your 2 bars. If you have got repeats, you can then work out the standard deviation and plot 2 times the standard deviation. If they didn't have time for repeats, you could pull the class data and they can still then use all of the classes data because they're following the same method to use that to calculate a mean, have your standard deviations or do a student t-test to see if there was a significant difference in the time it takes for tube X and tube Y to change colour, to lose that blue colour of the DC pip and become the same colour as our standard. So that is it for required practical 8. And don't forget you can download the full guide for free for this practical. And don't forget to hit subscribe so you don't miss out on any of the next videos. In this video, I'm going to be going through required practical 9, the respirometer for A-Level Biology. And we're going to be measuring the rate of respiration. Now I'm going to be talking you through the equipment, method, and how you'd collect these results and then how you'd process them. And if you want all of that in a free guide, I've linked that in the description below. Don't forget to hit subscribe as well because I'm going through all of the required practicals so you don't want to miss it. A big thank you to Woodford County High School as well for having me here today to film all of these required practicals for you. Now this practical itself isn't actually very complicated. It's quite easy for students to do. However, it notoriously doesn't work. So I'm going to be very surprised if I manage to get this to work today and we do actually see any coloured liquid move, but fingers crossed, let's see how it goes. So what you have here is your manometer, which makes up the pressure chamber part of your respirometer. I've got a clamp stand and clamp holding this in place. And then on the other side of this, I've got my 2 boiling tubes. This one here has got yeast. This one here has just got soda lime. I'm going to be adding 5 millilitres of glucose solution to the yeast, and I'm going to be adding 5 millilitres of distilled water to the soda lime. And we should find that we only have respiration occurring in the side with the yeast because it's a living thing, whereas soda lime is not living. So we should have just respiration on this side. We've also got these 3-way taps at the top connected to the bungs so we can control exactly where air can and cannot enter and exit these 2 boiling tubes. They're both going to be in a water bar to control the temperature, and we've got a thermometer to control that temperature as well. So in terms of risks linked to this practical, it's quite a low risk one. The main things is that yes, we are using yeast, which is a microbe, so wash your hands afterwards. And also we do have glassware, so there's always a risk that that could break and cut the skin. So make sure you don't have anything close to the edge. Boiling tubes, when they're not in use, make sure they are in a boiling tube rack as well. So let's just go through the theory of this experiment and explain what we expect to happen. So we've got the yeast on this side, and I'm going to be testing it under aerobic conditions. So there is oxygen already within those chambers. Now I've got the soda lime on both sides. The point of the soda lime is that absorbs carbon dioxide gas. And in aerobic respiration, when that yeast is respiring, it's going to be taking in oxygen, so therefore decreasing the volume of gas in the chamber because it needs the oxygen for respiration, and it's going to be producing the carbon dioxide. But that carbon dioxide isn't then going to stay in the chamber as gas because it's absorbed by those solid soda lime particles. So that means on this side where I have the yeast respiring, we should have a decrease in the volume of gas and therefore a decrease in the pressure of the gas on this side compared to the other side. And that should then mean that the coloured liquid is going to move down that pressure gradient. This side is higher pressure, this side is lower, so the coloured liquid should move around. And then we can measure the distance that that coloured liquid moved in a set amount of time. We can use the distance to work out the volume of gas absorbed in terms of oxygen, and then by looking at the volume of oxygen produced over time, so per minute, that can be used to estimate our rate of respiration. Now, the reason I say it notoriously doesn't work is all of that theory and setup, because this is so dependent on pressure changes and even atmospheric pressure, you sometimes don't see this coloured liquid moving. And when I say sometimes, you rarely do. And if you have managed to get this to work, please write in the comments what you have done to get this to work, because I've not spoken to any teacher who has managed to have guaranteed success repeatably. So let's have a go. We have got our yeast already measured out, the soda lime in both sides. Next I'm going to do then is set up my water bath. And I'm going to go for 37 degrees C because that's going to be the optimum temperature for the rate of respiration, and we want this to work. So the equipment then, we've talked about the clamp stand, the clamp, we've got our respirometer, 2 boiling tubes, 2 beakers, thermometer. We've got our yeast, soda lime. I've also got here my glucose glucose solution, a stop clock, and I've got syringes to be able to add our distilled water and glucose solutions to the appropriate tubes. So water bath, I'm going to add in some warm water, making sure it does actually cover the tubes. See what temperature we are at. Okay, that was at 40. Okay, they're both actually at 40 already, so that's good. We'll go for 40. Now we need to add our distilled water to this side, and we need to add our glucose solution to this side. But we need to be very conscious of the position of the three-way taps at this point. Now you want to leave this for about 10 minutes to acclimatize. And by that we mean reach this temperature, allow those chambers to fill with gas, let the gases expand as well, because this is all to do with pressure changes. And also for the yeast to reach that current rate of respiration. So we do need to get it all set up, leave it for about 5 minutes, and then we will measure it as we go. Now, while we're doing that and leaving it to acclimatize, I'm going to close the three-way tap to that chamber so that those gases in the chambers are expanding and it's reaching temperature, but it's not going to be impacting our results here. So now I've got those three-way taps set up, I'm going to add in my glucose solution to the yeast and my distilled water to the soda lime. Equal volume for both because we're using the distilled water as a controlled test tube. You do have to be very careful because this is all very delicate and fiddly. Okay, and let's leave that to acclimatize. So now we've left it to acclimatise and I can see it's bubbling away. We can now open up the valve so the gas is now entering into the chamber as well. And we've got these screwed on, so let's open and open. And you need to record the start position and start your stop clock. And that's at 5.3. So my start point here is 5.3. I'm going to leave it for 5 minutes and we should see the coloured liquid moves up this way. As you can see, it clearly hasn't worked, and I think our yeast has potentially been overactive and it's managed to bubble through. So that could be that maybe we didn't have enough soda lime in there to absorb the carbon dioxide that it was producing. And so there's now so much carbon dioxide, it's all bubbling over and it's going into this one. But here is what you would have expected to have seen. So to then work out the rate of respiration, we are going to work out the volume of oxygen taken up per minute. So let's say we did leave it for 5 minutes and we recorded the distance that coloured drop of liquid moved. We will then know the volume of oxygen taken up, because if we can work out the volume in that cylinder of oxygen that has moved into the yeast, we can then calculate it. So the diameter of that capillary tube, we could then use for pi r squared to work out the area of the circle. And then the distance that the coloured liquid moved, we times it by that, our length or distance, and that tells us the volume of oxygen that was taken up. And if we left that for 5 minutes, we would do the volume of oxygen taken up divided by 5, and that then tells us the volume of oxygen per minute. So if it was centimetres cubed, it would be centimetres cubed per minute as our unit. And if you were then going to do a comparison to see the effect of temperature on the rate of respiration, you could do that exact same setup, but instead of setting up at 40 degrees C, you could try a range of other temperatures and compare the rate of respiration. So that is it for Required Practical 9. Don't forget to click the link in the description to download the free guide for this practical and hit subscribe so you don't miss out on any other of the required practicals coming up. Hiya, and in this video, we're going to be going through the choice chamber required practical for A-Level Biology, and we're going to be using maggots. I'm going to be talking you through the equipment, method, risk assessment, tips and tricks for the students to make sure that they can get the best set of results possible, and the sorts of things you might want to show them and explain as you go through. And if you want all of that compiled as a free guide, then I'm going to link that in the description below. A big thank you as well to Woodford County High School for having me here today doing all of this filming. And don't forget to hit the subscribe button so you don't miss out on any of these required practical videos. There are lots of different options of how you could do this required practical. You can actually do it as a maze or a choice chamber. I'm going to be doing it as a choice chamber today, and I'm going to be using maggots. You could also use woodlice for this. Even within doing it as a choice chamber, there are different ways that you can do it. I'm just going to be talking you through one of the ways that it could be possible. So here we can see the equipment that I have. I've got a balance here, which you can use if you want to say a mass of maggots you're going to add, rather than saying an actual number of maggots. Here I've got my live maggots. I've got a stop clock, which I'm going to be using to make sure that the maggots are left for the same amount of time every time. So we've got that as a controlled variable. I've got spoon and pot and also forceps, all as different tools to help to transfer the maggots without causing any harm. I then have my actual choice chamber. So I've got my choice chamber and I've got labelled the conditions I'm going to have at the bottom. I have a paper towel, which I'm going to dampen with this water to put into the sections, which are the damp sections. Then once I've done that, I've got a bit of mesh here to put over the top so that the maggots can easily move over the top of that. Once I've then put the maggots in, I've got another piece of mesh to put over the top of that before placing the lid on. Now, the reason for that is the first piece of mesh is to stop them escaping from the bottom and so they can move more easily. The top one is to stop them from escaping from the top, because they could come out of that part otherwise. And then I've got this pre-cut out section of black paper that will slot over the side that says dark and dark to make that our dark conditions. So in terms of the risk assessment then, one of the key things is the handling of the maggots. There's two considerations here. First of all, you could get potentially microbes from the maggots, so you'd need to wash your hands thoroughly at the end. But also we need to consider making sure that we're not going to do anything that's going to cause harm to the maggots themselves as well, so handling them with care. And that's why I've got the plastic forceps to gently handle them, but also these plastic teaspoons. The next one is quite a low risk one, but we do have water, so if that spills, you could have people slipping, but also be aware you are in a lab, so if you've got water, that could spill on electrical items as well. So you need to make sure that water is not near the edge, and you do have paper towels nearby so you can mop it up if needed. The final consideration is the cross-contamination between repeats. So in between repeats, we should just wash it down, brush it down to make sure that there isn't any contaminations from the previous maggots on the mesh. That is actually going to help make sure that the trails that they might leave don't impact our results. But in terms of a risk assessment, it helps to prevent any cross-contamination between the organisms as well. So we need to set up our 4 chambers to start. We've got light, we've got dark, and we've got the damp sections. So I'm going to get one piece of paper towel to go into both of those damp sections, and we need to be adding the same amount of water. So these are the things to consider for the control variables. So I'm just going to fold it so that it can fit. So I've got one piece of paper towel in there. I'm going to get my next piece of paper towel in on this side, and then we need to add the same amount of water to that same size piece of tissue, just so we're keeping that controlled. Now you could be far more accurate with this. I'm just going to say I'm going to do 1 level teaspoon onto each. If you wanted to, the students could use a syringe or a pipette to make sure it is a more accurate measurement of that. I'm just going to make sure that is fully damp all over. So we've now got our 2 damp sections. If you do have silica beads, you can add those into the dry section. I'm not going to be using those, so I don't happen to have them, but you could put those there. Then we put on the first piece of mesh, and that is so that the maggots can't escape from the bottom. And now it'd be time for me to measure out the maggots. So this is where you could get the students to consider for themselves which would be better. Should we measure the mass of maggots and use the same mass of maggots every time, or should we use the same number? It might depend on what you're actually investigating. The key things though is they should be using the same age maggots every time, same species, because these could affect their movement and also the taxis and kinesis depending on what you're investigating. When the students have measured it out as well, they'll need to place it into the middle of the choice chamber, and And that's so they've all got the same start point, equal distance from all of the chambers. I'm going to leave the maggots there for 5 minutes. So they've got 5 minutes to make their decisions. And then depending on whether the students are investigating taxis or kinesis, you may just want to leave it set up 5 minutes. When the 5 minutes is up, get the students to take a photo. So they've got that snapshot of which chamber the maggots are in, and they can count how many there are in each chamber. That's what I'm going to be doing, so I can then calculate a chi-squared statistic afterwards. But they could, if they were going to investigate kinesis instead, they could have it recorded, or they could have paper where they trace the movement of an individual maggot, so you can actually then see the movement of it. We're going to go for taxis. We're going to see how many move into each section. So now we've set up those different conditions in the chamber. You should leave it for about 5 minutes so that it does acclimatise. So we do have those sections that we said are damp. There is time for that air to become damp. The sections that we've said are dry, particularly if you've added silica beads, they are now going to be dry. Sometimes you might also want to say you're going to add your maggots so that they have time to acclimatise for 5 minutes before you record the results, or you could just add an extra 5 minutes onto the time that you're measuring for. So you could say 10 minutes, I'm going to leave them for instead of 5, and that will take that into account as well. So I need to get my maggot lid open, and this is when you then need to be really careful that we don't get escaping maggots. Whilst also considering the ethics of this investigation, we are using a living thing here, so we don't want to be causing any harm to the maggots. As you can see, I've already lost one, so you do need to be really, really careful. I'm actually going to go for number as well as mass, so I can just compare that each time. So if there are any differences each time, we can see was the mass actually different. So I'm going to count out 10, but also measure the mass whilst keeping an eye on these maggots that are trying to escape. Students can wear gloves if they want. I'm choosing not to, but I'm going to be washing my hands at the end. So I've got 1, 2, 3, 4. Obviously I do have a little bit of sawdust in there. 5, 6, 7, 8, 9, 10. Okay, so that's my 10. So I do have a few runaway maggots. So 1.01 grams, that is the mass. I'm just going to double check we do have 10, which is quite hard when they're wriggling. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. Yep. So now I would place them in the middle. You can already see they're starting to move. Place that on top so they can't escape. It is going to be tricky for the students. Now, the idea of this hole is you could actually just put them in there straight away. So you can see mine are already moving before I'm actually ready. But if you do that, you then can't put the mesh, but it's your decision which option you go for. I'm going to go for this, press start, and now leave that for 5 minutes and see which chamber most of them end up in. So my time is now up, so I'm going to actually record my results. The main tip I have here for the students is, because the maggots are constantly moving, when you take off that black bit of paper, get them to take a photo and then they can count from the photo. I'm going to do it live, but it would be better to do it from the photo. All right, so I can see 1, we've got 1, 2, 3. So that was damp and light. It was 1, there was 3 here, which was damp and humid, none in that one, and then that must mean that there was 6 there because I added 10. Okay, so that is my first trial. Now what we'd need to do is take those maggots out, give the mesh a little bit of a clean, and we'll run it again. I'm not going to repeat all of that because we've seen how we've done it, but we can just have a little look at how the students can then carefully return the maggots. They will need to move quickly with this because they're going to be pretty quick movers. Okay, so that's my 10, but as we can see, we do have residue here. So we'd need to just clean that in between. They could just take that to a sink or a bin and just shake it down and give it a quick wipe. Once the students have repeated this at least, so they've got 3 trials so they can calculate a mean and discard any anomalies, then we can start to think about how we can use the chi-squared statistic. So in our chi-squared statistic, I'm only going to go through the brief information here. If you do want a full video on how to calculate the chi-squared statistic, then I'll link that in the description because that's actually over on my other channel, Estruch Biology. But the key thing is here, we added 10 maggots. When it comes to the chi-squared statistic, our null hypothesis would be along the lines of there'll be no difference between the number of maggots that we expect in each chamber and the number of maggots we observe in each chamber. And when we say the number we expect, we'd be expecting an equal split for our null hypothesis. So if I've added 10, that would mean 2.5 per chamber. Now we can't actually get 2.5 of a maggot, so maybe you might want to get your students to use 16 maggots, and then it could be 4 per chamber and it works better. But in terms of the maths, saying 2.5, it's fine for the statistic. Then we would look at our mean to see what we actually observed in each of the chambers. And once you've got your observed and your expected, you can then use this formula to calculate your chi-squared value. They can then compare it to the critical value for the 3 degrees of freedom. 3 degrees of freedom because we had 4 categories, -1, 3 degrees of freedom. So their critical value would be 7.815. So then have a look, was their calculated value the same or higher than the critical value, to be able to conclude whether there was a significant difference between what we expected and we observed at p equals 0.05. As I said, full explanation on the statistic is in the video I'll link below. So finally, some top tips for success that you could share with your students. Number 1 is making sure they are using a consistent number of woodlice every single time. Number 2, make sure that they do have a minimum of 10 minutes in the choice chamber, because then that gives them 5 minutes to acclimatise, 5 minutes to move around. Number 3 is making sure that everything is set up in the choice chamber and ready to go before they add their maggots to the centre, so that there's no delay in adding the maggots, and therefore you can start straight away. Number 4, making sure they are handling those maggots with care, not causing any harm, and working quickly to make sure they collect any that might escape. So that is it for this required practical, and don't forget you can download the entire guide of this required practical linked in the description below. But that is it for this one. Hope you found it helpful, and I'll see you in the next. In this video, I'm going to be going through required practical 11 for AQA A-Level Biology, which is preparing your glucose concentrations with the dilution series and then using your colorimeter to create a calibration curve. And you can then use that calibration curve to identify unknown concentrations of glucose in a hypothetical urine sample. So we're going to go through the equipment, the method, do the actual experiment, how you'd present the results, and I'll tell you some of the top tips for success to share with your students. And if you want all of that information in a downloadable guide for free, it's linked in the description. Don't forget to hit the subscribe button, because I'm going through all of the required practicals and you're not going to want to miss them. And a big thank you to Woodford County High School for letting me film here today. So I need my stock solution of glucose and distilled water to create my different concentrations. I also have a range of syringes to use for this. So I have a selection of 1 milliliter syringes as well as 2 milliliter syringes as well. I've got my test tube rack with lots of different test tubes, well, 6 different test tubes to create those different dilutions in, and a permanent marker to label these to make sure they do not get confused, because they're all colourless liquids at this stage. I've got my quantitative Benedict's solution, stop clock, lots of dropping pipettes so I can transfer my solutions at the end into the cuvettes. I've got my cuvette holder, my colorimeter, and I've got a large beaker and a thermometer to create my water bath for the experiment. So before we get started then, you need to make your students aware of the risk assessment. Now, first of all, in order for the quantitative Benedict's to work, we need to be using water at 90 degrees C. So I'm going to be using water from a kettle to avoid having to have Bunsen's, because the flame is an extra hazard, but the water itself could still cause burns, so they need to make sure they are working really carefully. The quantitative Benedict's as well is harmful to the environment and an irritant, so it must not go down the sink, and they must wear goggles the entire time. The glassware could smash and cause cuts, so it's really important that we use a test tube rack to hold all of the test tubes, not leaving any on the table so they could roll off and smash. And then the final one to be aware of is our colorimeter is plugged into the mains and we have liquids here. So any spills must be cleaned up straight away so we don't get any liquid onto the plug socket. So let's get into the method then. And the first thing I'm going to do is label all of my test tubes. So these are the different concentrations that I'm going to be making. So that's what I'm going to label. I've got 0, 2, 4, 6, 8, and 10. I'm going to put my distilled water into this beaker, because otherwise my syringes won't be able to fit into this bottle. So I'm going to add in water to all of them first, and this one here is 2, it's just water. So I'm going to add in my 2. That one is 1.6. So I'm actually going to swap over to these now to get more accurate measurement. 1.6. That's 1, and then this one will be plus 0.2. Next is 0.8 of water. And then 0.4. And finally, no water. So next I'm going to do the same thing and pour my pre-made 10 millimoles of glucose solution into the beaker so it's easy to get out. Just going to give it a shake before I use it so it's definitely all mixed up. Get a new syringe. So this one here is 2 of glucose. The next 1.6 and 1.2, and this one will be 0.8 and then 0.4. I'm then going to get a bung to put on top because we need to mix all of them so that we do have an equal distribution, so we definitely have the concentration we're expecting throughout. So next on our list is we need to place all of those test tubes into a boiling water bath. I'm not going to completely fill it because when you add the test tubes, that displaces the water, so it's going to, oof. I was not expecting that. Don't want the bungs on anymore because we've mixed it. Okay, let's do that again. So next then I'm going to put the test tubes in but take the bungs off because as they go into the hot water that can cause an increase in pressure in the test tubes and make them pop. Oof. Was not expecting that. And we'll leave them for about 4 minutes to reach temperature. So now we've got those mixed and we know they are equally distributed. I'm going to add the Benedicts. Now so that it can actually fit in, I'm going to go for 2 centimeters cubed, just using my dropping pipette into each of them. Then I'm just going to mix it. And now we're ready to put all of those into our water bath. And it'll take probably about 4 to 5 minutes for the reaction to occur. I'm actually gonna boil the kettle again because it does need to be around 90 for this to work, and I can see it's already dropped. So that's now had long enough, but we need to let it to cool for 5 minutes before we put our samples in the colorimeter. So I'm going to take them out and being very careful not to shake. So any precipitate that's formed will start to settle at the bottom. We'll leave it for about 5 minutes, but for this one we can basically just touch it to make sure it's not going to be too hot to go into the colorimeter. So we're now ready to transfer our solutions into the cuvettes for the colorimeter. So with the colorimeter, some of the key things that you need to make your students aware of are, first of all, they need to zero the colorimeter before they start. So you need to make sure that you have a cuvette filled with distilled water to use for this. They also need to make sure that the color filter that they're using is the opposite color spectrum to the color of the solution. So this is blue, so I'm going to go for red. So then I'm going to put my cuvettes in, making sure that I'm only handling it on the ridged side so I don't leave any grease and fingerprints on the front. And you've got the frosted triangle at the front. Students sometimes don't push it all the way in. You need to listen and feel for the click. And then press R to reset to say that is our 0. That is our standard that we are comparing to. I'm then going to use a different drop-in pipette to transfer the solutions into each one. And be very, very careful because the precipitator settled at the bottom and I don't want to draw that up. And I want it about 3/4, or at the very least half full. So that should be fine. So now we're ready to test the absorbance of each of these samples. And I've got my table to record the results as I go, making sure I'm holding on the ridge side and frosted triangles at the front. When I push, make sure it does click, and we press T for test. So that was my 0, so 1.15. And then my 0.2. I've actually got slightly different concentrations to these, but I think it's because I've said millimole here, so yeah, that's fine. 1.32. 1.46. 1.54. And 1.24. So we can see that we do get this nice increase up until the 1.24 where it drops. So as we saw, the absorbance actually dropped when it got to the highest concentration. So I then went away and did some research, and here are some of the reasons why. First of all, it could be the Beer-Lambert that only works in a linear range. So at very high absorbances, the colorimeter can't measure accurately, so the values flatten or even dip. Secondly, maybe the Benedict's reagent can become limiting. If there's more glucose than copper ions available, there's no extra products to form, so the absorbance stops increasing. And thirdly, it could be maybe large clumps of precipitate are scattering the light differently or settle at the bottom, actually lowering the reading. So to fix it, you could try adding excess reagent or more reagent to each one. Have a little go, but if you have found anything that helps with this, then let me know in the comments. So now you've got your results, your students would plot the absorbance on the y-axis, concentration of sugar on the x-axis, plot their data and create the calibration curve. And you could then give them the unknown glucose concentrations, which are our fake urine samples. They would do exactly the same thing again. You'd have your unknown urine sample, add the same volume of Benedict's, place it in the water bath for about 5 minutes, let it cool down for 5 minutes, put it into your colorimeter, measure the absorbance, and then on this graph here, we can see whatever their absorbance was. And in this example, we've got the urine sample had an absorbance of 0.58. We then read off to see where it intercepts our line and read down to see what glucose concentration that is. And we can then do that to work out the glucose concentration in the urine sample. So some top tips just to make sure your students are aware of in this investigation. Make sure that they do label their test tubes right at the start so they don't get them confused when they're all colourless liquids. Make sure that they do always zero their colorimeter using distilled water at the start. Make sure they're not touching all the sides of the cuvettes, making them greasy to affect the light absorbance, and that they're definitely placing it into the colorimeter with the frosted triangle facing forwards. And then with regards to that quantitative Benedict solution, it does work best if it's fresh, and you need to make sure that you are adding the same volume every single time, because that's going to affect the amount of precipitate that would form. So that is it for this required practical. Don't forget you can download the guide for free, go through all of this experiment, and don't forget to hit the subscribe button so you don't miss out on any of these videos. Hey everyone, and in this video I'm going to be going through with you the sampling required practical for AQA A-Level Biology, but it is actually on all of the A-levels, and it comes up in the GCSE as well. So we're going to be thinking about the equipment, method, risk assessment, and how you can collect that data, plus what the common mistakes students make and top tips you can share with them. And if you want all of this included in a free guide, I'll link that in the description for you. If you are new here, don't forget to click subscribe, because I go through all of these required practicals, plus so much more to help students. And a big thank you to Woodford County High School for letting me film here today. Now I'm going to be going through with you both random sampling and a belt transect, but generally speaking, the equipment that you will need are, first of all, either you're going to need your gridded quadrat or you're going to need an open quadrat. I've got 2 tape measures, so I can use these 2 tape measures lined out at angles, and I'm going to be using that in my random sampling, and then just 1 tape measure when it comes to the belt transects. I've got a meter ruler here, but I don't think I'm actually going to use that for what I'm doing. But if you were going to be measuring, let's say, the height of plants or something, then you could use a meter ruler. And also I've got a light meter so that I can measure the light intensity in the different areas. Final thing that you might need is a key for students to help to identify the plants. So the risk assessment for this required practical is going to vary depending on the time of year. I'm here filming in autumn. It is pretty cold and I do have a coat, but I've sacrificed it for the filming quality. But in terms of your students, the key things for the risk assessment are considering the weather conditions. So do they need several layers to wrap up warm? Do they need to consider dehydration, heat stroke, sun cream, sun hat? So considering the weather. Then you need appropriate footwear, because you've got lots of uneven ground and it might be muddy and slippery. So they need to have appropriate footwear to reduce the risk of them slipping, twisting their ankle. At the end, they should be going to wash their hands, because we never know what's actually been on the ground. There could be all sorts of microbes, feces, so washing the hands at the end. And there could be the risk of plants that could sting. There might be nettles. Students might have allergies, hay fever. There could be insects that could sting as well. So those are the other considerations that students should have for their risk assessment for this required practical. So which quadrat should students be using? Well, it completely depends on what they're going to be measuring. If they're going to be recording the abundance of a plant species where there is far too many to count accurately. So for example, the grass. I'm not going to be able to count every single grass plant in this field accurately. Or if it was moss where you can't completely tell where one plant starts and the next ends, then it'd be better to do percentage cover. And that's when you want to use a gridded quadrat. So you could place the quadrat down and students can estimate how many of these squares are at least 50% covered by the plant. And if it's at least 50% covered, we'll count that as 1%. And the reason we're saying at least 50% covered is that works as a way to standardize the way we are estimating percentage cover. If they're going to be estimating the abundance of a plant species where you can easily identify one species and there's not too many to count, then you want to be recording density, which means literally counting how many of that plant is in your quadrat. And to do that, you're going to want an open quadrat so you don't have anything blocking your view. So we're going to go through the method for random sampling, and I'm going to go for clover, I think, because looking around, I can see there is quite a bit of clover. Now, this required practical is the most assessed required practical. It's come up every single year, probably because it's a required practical, but it also appears in the theory. So I'm going to go through this method with you now. Number 1, the first marking point is always create a gridded area. And the way you can do that is lay 2 tape measures out at right angles. So here we go. So that's mark number 1. We've got our gridded area. Mark number 2 is then how you create your coordinates. And you need to use a random number generator to create your coordinates, which you can just do on a calculator. You could do it on a phone, but the marking point is going to be to create coordinates. Once you've got your coordinates, let's say I've got 3 and 2. Ideally, you would do it in pairs, because then one of you would go 3 meters along and the other would go 2 meters up. And where those coordinates meet, that's where you place your quadrat. So let's have a look. 2 here. And 3 is just there. So now I'd place my quadrat. So then you need to encourage the students to actually get down to the ground so they can accurately record what they have. You'd need to look through, and I've got so much leaf litter here because I'm doing this in autumn and that doesn't count. So I need to try and move all of that out the way. And then I can see here, I've got some clover here. As I said, it's got to be at least 50% covering a square to count as a percentage. So 1, 2. So that would be about 12%. So I've now worked out that for one quadrat, I've got 12% in terms of clover coverage. If you were then going to measure any other variables, so I have a light meter here, I could record that every site as well to see later on, is there any correlation between the light intensity and the percentage cover? So for this one, I need to hold it out, making sure the sensor is exposed to the light, hold it still, and then I can record down my light intensity. So that's marking points 1 and 2 that they need to know. Next then, the marking point would be repeat and get a large number of samples of quadrat. Now on the mark scheme, they varied this. They used to say it has to be at least 20 quadrat samples. More recently, they've said 10. Because they've changed their mind, I would stick to them saying a large sample because they've always allowed that. So collect a large sample and then the students would have to calculate their mean percentage cover. The mark scheme has started to insist that you actually describe how to calculate the mean as well. So add up all of your percentages, divide by the numbers of samples you took. Now, if you had been collecting density, there is a final mark that you'd have to get your students to include as well. And that's because percentage, that is comparable for whatever your mean percentage is on your quadrat, then that is going to be the same mean percentage for the entire field. However, if your students have to calculate the density, so the actual number of a plant in the entire field, if we've done our large sample, we calculated our mean, and we had, let's say, 5 daisies on average or as our mean in our quadrat, we then need to scale that up for the entire field. And students need to know how to do that. That's the final mark. So they would need to know how many quadrats would fit into the entire field. And your mean for one quadrat, you times that by the number of quadrats that fit into the field. Now, the other option of sampling would be a belt transect. And this is if you want to see the change over a particular distance. So for example, here, if I wanted to see what is the impact of that path on the percentage cover of clover. And it might be because I think we'll get more or less clover closer or further away from the path. So everything I said about the quadrat, that's exactly the same. Large number of samples, calculating the mean, that is all the same. But the key things that are different then is we're not setting up 2 tape measures at right angles. We're not doing random sampling. And we are going to be doing instead systematic sampling. So that means starting at 0, I would place my quadrat at 0. And the tape measure is set up at a right angle to what we think is going to be potentially causing the change. Whether that is, like in this case, a path or a river, we have it perpendicular to what we think is causing the change. And then you'd place your quadrat at intervals along your tape measure or our transect. So I place it at 0, estimate my percentage cover, and then I might move along 1 meter, 5 meters, whatever is appropriate. And the way to decide what is appropriate is how quickly do we see a change in the plant distribution. If it looks pretty similar all the way along, then you might want to do it every 5 meters. If it looks like it's quite drastically or rapidly changing, then you'd want to do it every meter. So that's what the students would need to say. You'd place your transect at a right angle to what you think is causing the change or that you're investigating. You'd then place your quadrat at intervals. You would do a large number of repeats again, but what that means this time is you would take another transect, place it in parallel and record it for all of those positions again, and then the same along here, and you do that a large number of times and work out a mean for every position on your transect. So that is it for required practical 12, the most commonly assessed practical. The key thing your students need to know are the 4 or 5 marks for a random sampling method, a belt transect method, and also thinking about risk assessments as well. Don't forget that if you are new here, hit subscribe so you don't miss out on any of these videos, plus the free guide is linked below.